Hello everybody and welcome to the Cooper Smith Career Consulting Crash Course on Introduction to Life Processes. SCI 102 is the course number. It's basically biology. We are going to be focusing on the life processes specifically. We're going to be discussing general life processes, and then we're going to be applying it to the life processes as they are applied in the human being. Other courses might deal with animals and plants and things like that. We're not really going to focus much on animals and plants except maybe a little bit tangentially, but we are going to be focusing mainly on the human being and the life processes. But before we even get to the human being, we'll have to discuss the background of what they are and what they do. There are various textbooks and resources, study guides that you are provided with, of course, but I'm not really going to be focusing on that in this video. I really intend this video to be kind of a standalone review that will give you the ability to be prepared for the final examination, generally speaking, by just reviewing whatever material there are in this course. This PowerPoint presentation over here that I'm going to use, you should have access to. If you don't, you should ask the appropriate person that's helping you with the course. And we are going to now begin with our discussion of life processes. Our first topic that we're going to cover is a little bit of basic chemistry. Of course, this is not really a chemistry course, but a little bit of basic chemistry is really necessary for biology. This unit, this section, is atoms, molecules, and life. Life is based on, well, everything is built from molecules, and molecules are built from atoms. A molecule essentially means the smallest thing in nature that cannot be broken down and still remain the same object. You can have one molecule of wood, you can have one molecule of water, you can have one molecule of iron, you can have one molecule of steel. If you break down the molecule any further, it's not going to be water or steel or whatever it is. You can break it down, but not retain its structure. The atom is the building blocks of molecules. Atoms in general, hopefully you might be familiar with it, but all the atoms that are in nature are based on elements. In other words, one atom means the smallest unit of a particular element. All of the elements are listed on the periodic table of the elements. You may have seen this before. This is unfortunately, I can't get the entire periodic table in the view over here, so I'm going to have to scroll around a little bit. But you can see it starts all the way at number 1, which is the H, which is hydrogen. Then it goes to helium, then lithium, beryllium, and I'm going to move back over here to the other side of the periodic table: boron, carbon, nitrogen, oxygen, fluorine, neon, etc., all the way into the low 100s when it comes to the elements. If this were a chemistry course, we would go through these in exhaustive detail, and we would discuss what the different properties of different atoms are and how they work, different elements are and how they work. We're not really going to do that because that's not really necessary, but I do want to show you a couple of things that are very important. Really, this number over here on top of each element is the number of protons that are in the element itself. There are 3 types of basic particles in an atom. There are the protons, neutrons, and electrons. The way an atom basically looks is that there is a material in the middle, those blue and red things in this particular diagram, and then there are the things swirling around the middle, and those are the black things in the diagram. The middle is called the nucleus, although in this diagram it looks like the nucleus takes up a large component of the atom, when in fact the nucleus is only a tiny, tiny, tiny little thing in the middle of the atom. But the nucleus, the middle things, the blue things and the red things, are the protons and the neutrons, and the things that fly in the outer circles, sometimes known as orbitals, are the electrons. Those are the black things. The protons are positively charged, the neutrons have no charge, and the electrons are negatively charged. In a typical atom, in its ground state, in other words, in its normal state, the number of protons is equal to the number of electrons, and therefore the atom is neutral. It has no charge overall. It has a certain number of positives and an equal number of negatives. If there are more electrons than there are protons in the atom for whatever reason, then the atom has a negative charge. If there are more electrons than protons, then it is negatively charged. If there are more protons than electrons, then it would be considered positively charged. Either way, as we'll discuss in a couple of minutes, that is what is called an ion. An ion is a charged particle. In the periodic table, this number above the name, above the letter that represents the element, H for hydrogen, Li for lithium, sodium, potassium, calcium, magnesium, all these other things over here. This number, this 1, this 3, this 4, this 11, 12, that represents the number of protons in the atom. So an atom with 1 proton is hydrogen. An atom with 3 protons is lithium. An atom with 11 protons is sodium. As we move to the other side of the periodic table over here, an atom with 8 protons is, of course, the very important element of oxygen. An atom with 17 protons, for example, is the element chlorine, etc. The important thing is that in its normal natural state, they would have the same number of electrons. For example, fluorine, which is F, which has 9 protons, would normally also have 9 electrons. If it doesn't, it would be considered an ion. Because ions are atoms with a charge. Atoms, the way this works in terms of charges, is that the number of protons stays the same. Remember, the idea in ion is when you have a different number of protons as electrons. Theoretically, there would be 2 ways to do that. Either you could add or take away protons, or add or take away electrons. Problem is, you can't do that with the protons. If you change the number of protons, it wouldn't be that element anymore. If you had, for example, an atom of potassium, which is K, which is 19 protons, and you changed the number of protons, it wouldn't be potassium anymore. If you added a proton, for example, you'd have calcium. If you added 3 protons, you'd have Ti, whatever that stands for. If you took away a proton, well, then you'd have Ar, which is the 18, and you'd have argon. So you do not take in, while that's theoretically possible in certain ways, you don't usually create an ion by changing the number of protons. Instead, you change the number of electrons. So for example, if you have sodium, which is 11, and you have 11 electrons, which is a neutrally charged sodium atom, if you add an electron, well, now it is negatively charged. If you subtract an electron, well, now it is positively charged. Now you'd only have 10 electrons, and you'd have an overall charge of +1. Take hydrogen, for example, which has 1 proton and 1 electron. If you take away the electron, well, hydrogen is now +1 charge, because it's got 1 proton and 0 electrons. If you added an electron, well, now you'd have a charge of -1, because you'd have 1 proton and 2 electrons. The next thing that's important to know about about elements and molecules, is that elements do not tend to stay only with each other. Atoms tend to bond with other atoms of different elements. If you, for example, never had any bonds between different atoms, then the only substances that would exist would be the ones on the periodic table, things like hydrogen and chlorine and oxygen and things like that. But of course, we know that there are much more complex substances like quartz and water and sugar and all the things that we use and that we interact with on a day-to-day basis are actually many different atoms that are combined with each other. And they are combined through what are called chemical bonds. A chemical bond is, simply enough, a bond that combines 2 atoms together. There are 2 basic types of bonds. The differences between them are very subtle. We'll very, very quickly go over them. An ionic bond is essentially when you've got 2 atoms that are next to each other, and one of them kind of borrows the other atom's electron. Let's say, for example, you've got sodium and chlorine. Sodium is Na on the periodic table, and chlorine is Cl on the periodic table. We look over here, we've got sodium, which has 11 protons, And we've got Cl, which is chlorine. By the way, sodium and chlorine, NaCl, is actually sodium chloride, otherwise known as regular plain old table salt. When you pass the salt and put it on your bread or soup or whatever it is, that is sodium chloride. Now, what happens when you have a bond between sodium and chlorine, for reasons that I don't want to get into that you might know from your high school chemistry class or whatever it is, if you've taken it, or even elementary school chemistry class, sodium tends to lose an electron. Long story short, the reason is because the way the electrons are structured and the orbitals that they're on, it's more natural for there to be 10 electrons in an atom than for 11. A 10 fills an orbital, and the 11th is just kind of hanging out there all by itself. So sodium has a very strong tendency to lose an electron, and therefore become a positively charged. If sodium loses an electron, now it's only got 10 electrons and 11 protons, it becomes positively charged. Chlorine, on the other hand, has a very strong tendency the opposite. The number that fills up the orbital in the chlorine is 18. 18 is a much more comfortable number, so to speak, to have than 17. So chlorine, on the other hand, tends to gain an electron. And when chlorine gains an electron, it's got 17 protons and 18 electrons, and that makes it a negative one charge. Because of that tendency, sodium tending to gain an electron and chlorine tending to lose an electron, those two tend to come together in very strong bonds. So just to put it in other terms, the sodium is very happy to give up an electron, and the chlorine is very happy to take an electron. And that kind of bond is called an ionic bond, because they're both ions. The sodium becomes a positive ion, and the chlorine becomes a negative ion. Looking at this diagram, the one on the left over here is a normal natural state sodium ion, which has 11 electrons. The protons and the neutrons are in the middle over here in the nucleus, and then the electrons would be on the outer rings. And you got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. And notice the 11th one is all the way out here, all by itself on its own ring. That is not a particularly comfortable state for an atom, and therefore it can lose an ion. It can lose an electron, and go down to only two orbitals that have 10 electrons altogether. The chlorine, on the other hand, in a sodium chloride ionic bond, it starts off as the opposite. It starts off, remember, with 17 electrons. Here's the nucleus in the middle. It's got 17 electrons floating around the outside. And notice this outer wall of electrons, this outer orbital of electrons, is missing one. You got 2 over here, 2 over here, 2 over here, and 1 over here. That state is the reason why a chlorine atom will have a strong tendency to gain an electron. When you have this combination, the sodium atom, which is only too happy to give up an electron, combines with the chlorine atom, which is only too happy to get an electron, and what you have is the result over here. You have the sodium combining with the chlorine, so that the sodium is a positive ion and the chlorine is a negative ion, and the two things come together in an ionic bond, which is NaCl, also otherwise known as sodium chloride, otherwise known as table salt. The other type of chemical bond that exists, other than the ionic bond, is called the covalent bond. The covalent bond is similar to the ionic bond, except that instead of one atom giving up an electron and the other atom gaining it, the covalent bond is where the two atoms kind of share that last electron. Here's an example of a covalent bond. This example of a bonded molecule actually happens to be methane, which is CH4, 1 carbon and 4 hydrogens. This is an example of a covalent bond because the red circles are electrons from the hydrogen. Remember, hydrogen was a 1 on the periodic table and only had 1 proton and therefore 1 electron. Carbon happens to be a 6 on the periodic table, but the way carbon and hydrogen combine, and the way many other types of substances combine, is rather than the hydrogen giving up an electron and the carbon getting it, they kind of share electrons. One carbon electron shares or comes together with one hydrogen electron. So that's an example of methane. Here's another example. The next, really a big example, one of the most important examples we know of, and that is water. Water is H2O, which means 2 hydrogens and 1 oxygen. And they come together through this process of sharing the electrons. And hydrogen bonding also, hydrogen bonds are really kind of a subset of covalent bonds, but hydrogen bonds are attraction between hydrogen and another substance, such as nitrogen or oxygen. Water molecules, as we just discussed, is a very important type of chemical compound. It's made up of hydrogen and oxygen, 2 hydrogens and 1 oxygen. That's why, as you probably know already, hopefully know already, water is sometimes known as H2O, 2 hydrogens and 1 oxygen. This is the diagram that we just looked at. This is one water molecule on the upper left-hand side. Notice the oxygen is bigger because the oxygen has a number of protons of 8, whereas the hydrogens only have 1. This diagram is not really to scale. The oxygen is really much bigger than the hydrogen atoms. In this one, it looks like the hydrogens are just a little smaller. They're actually much smaller. But water is a substance whereby the hydrogens are bonded to each other. And that's why sometimes you'll hear water being referred to as being hydrogen bonds. And the reason is because the hydrogens of the different water molecules are bonded to each other. And because of that, water has many very important and very unusual properties. We're going to discuss some of these. And some of these are necessary for human life to exist. So we're obviously very happy that water does have some of these particular types of molecules. First of all, water has, like many other substances, cohesion, which means the tendency for molecules of a single type to stick together. Water especially has this element of cohesion, more so than, let's say, for example, sand. If you dropped a bunch of sand molecules, or not molecules, but grains, let's say grains of sand, they would scatter out all over the place. They would not bond to each other at all. Water, on the other hand, does. If you've ever filled up a cup a little bit higher than the cup would ordinarily hold, and you see there's kind of like a very small, low bubble in the middle. It's hard for me to describe it. Let me look for a picture. Okay, there we go. Found one. I'm sure you've all experienced this. This is a glass that you filled with water, and even though the water is actually a little bit higher than the glass, and you would think that it would just kind of spill out the side, very often the water kind of holds together and does not spill over the side. That phenomenon is known as surface tension, and the reason for it is the natural sticking together that water molecules do. All the water molecules stick together. Once you puncture it, like if you stick your hand in the top, then you might see the whole thing kind of spill out over the side because you broke the surface tension. That's also why droplets of water attached to something, like this example of some droplets of water on a leaf, which you've seen obviously an infinite number of times in various contexts, droplets of water stick together really for the same reason, based on this cohesion, this concept that water molecules stick together based on their hydrogen bonds. And this process also causes what's sometimes known as capillary action, which is very important for plants and leaves. We're not going to discuss it in too much detail, but basically the way it works is that water, when it goes into a root or goes into some kind of a filament, some kind of a hair on, it doesn't have to be a plant, it could be, pretty much anything, the water actually gets drawn naturally with the other water that it kind of gets drawn up the plant. Like, for example, the plant water enters the roots and gets drawn up the bark of a tree all the way to the leaves where they're needed. So in order for the water to reach the leaves, water fills tiny tubes that connect the root, stem, and leaves. Before water can evaporate, it gets pulled up naturally to all the way to the leaf surface. And that allows it to be pulled upward in a chain, which is a very unusual property. I have just, again, let me give you one quick example over here. I wanted to show you a little experiment that somebody did. I just want to show you how this works so you get a little bit of a view that just let me, well, 1 second. I'm sorry, I needed to find you a better experiment than that. The other one you couldn't really see very well. So this is just an example of capillary action that I thought was very interesting. The person puts the red water, a paper towel, really just a folded up regular paper towel that you can do in red water on the right, and a blank cup or an empty cup on the left. And look what happens. You've probably noticed this when you use a regular Bounty or some kind of paper towel, and you put it, and you put it on the floor, and it soaks it up. Even if you don't actually scrape it, you just kind of put it in the whole paper towel. Kind of draws the water, and the reason why it's drawing the water like that is because of capillary action. And of course, you can see over here, this is a time lapse. Doesn't happen really this quickly, but the water kind of gets drawn from one to the other until the two cups will actually level out. And so that's an example of cohesion and how that works. Surface tension we also discussed is really the same concept, the tendency for water to resist being broken, and therefore it is produced by this concept of cohesion that we discussed in the last few minutes. Another very important characteristic of water is the water temperature itself and how it behaves with its environment. First of all, water has among the highest specific heat capacities of any substance, which means water is very difficult to heat up and it's very difficult to cool down. I wouldn't say difficult, but it takes a long time and it takes a lot of energy. It's a lot easier, for example, to heat up metal quickly than it is to heat up water. If you take a pot or a pan and it's empty and you put it under a fire, it's going to heat up extremely quickly, whereas if it's full of water, it's going to heat up much more slowly. And the opposite is true. It's also going to cool off much more slowly than if it were empty, or if it were filled with metal or some other object that's not water. The specific heat of an object literally means the amount of energy it takes 1 gram to heat 1 gram by 1 degree Celsius. The specific heat of water is 1, which means it takes 1 calorie of energy to heat 1 gram of water by 1 degree Celsius. Whereas metal and other objects have much lower specific heats. Most metals, for example, have specific heats that are somewhere between 1/25th of that of water and maybe about 1/5. Aluminum is about 1/5 of water. Lead is about 1/25th of water. The point is the heat capacity, the ability to absorb heat is much, much lower when it comes to metal and pretty much every other substance on Earth than when it comes to water. That's why, by the way, in case you're curious, islands or places that get their weather from the ocean, like the west coast of the United States, where the weather blows in from the Pacific Ocean, tend to be much more moderate than places in the middle of the continent or places that are surrounded by huge swaths of land, like Siberia, where the temperatures get very high or very low, depending on whether it's summer or winter. This example is a pretty simple example. If you, as we discussed before, you put a pot of water on the fire, the pot is going to get hot very quickly. The water, on the other hand, is going to take much, much longer to warm up. The amount of water it takes to turn, there are really 3 states of water, just like every other object. There's the solid state of water, which is ice. There is the liquid state of water, which is water. And then there is the gas state of water, which is steam, or water vapor. The amount of heat needed to make a substance evaporate is called its heat of vaporization, and water has a very, very high heat of vaporization. It's about 540 calories per every gram. That means to vaporize 1 gram of water, you need 540 calories of heat, which is, a substantial amount of heat. And this is one of the reasons why when you sweat, or even when you splash water on your face, it cools you off. Normally, if you go outside and it's 95 degrees and you're running around a lot and you get very hot, if you wouldn't sweat, your skin would get extremely hot very quickly. The way sweat works, or the way water that you splash on your face works, is the hottest molecules. The molecules that are moving the quickest, which means the hottest molecules, they evaporate. They basically heat up the water that's on your face or that's on your arms, and they go ahead and evaporate. And what's left over is just the cooler molecules. The hottest molecules turned into steam and evaporated into the air, and the rest of them stayed on you. That's why and you can try this, if you're very hot and you splash warm water on your face, it's still going to cool you off. Maybe not in the first millisecond, but after a few seconds it's going to cool you off. And the reason is because you splashed even warm water on your face, but the hot molecules on your skin and the hot molecules that are floating around evaporated into the air, whereas the cold molecules, or the cooler molecules, so to speak, are the ones that are left on your face, and that's going to cool you. So, I mean, don't do this with scalding water, obviously, then you're going to hurt yourself. But you take warm water, 100, 105, 110 degree water, as long as it's not scalding and it's not going to hurt you, you try splashing that in your face, it's still going to cool you off after a few seconds, because the hottest molecules are going to evaporate and the cooler molecules are going to stay there. Other interactions with molecules, there are a few other things that are important. First of all, water is what's sometimes referred to as a universal solvent. Water is one of the best ways to break down molecules. Some things break down more easily than others. Substances that break down are called substances that break other things down, that allow other things to be broken apart, are called solvents. Water is an excellent solvent. For example, when you put salt in water, it's no longer the same molecular salt. Instead, you get, well, as we all know, you get salt water. Sugar also. Now, sugar doesn't dissolve quite as well as salt does, but it also dissolves in water. You take your morning coffee, even if it's cool, even if it's iced coffee, you put it in a packet of sugar, you stir it around. Maybe not every grain of sugar is going to dissolve. Some of them are going to stay on the bottom, but a lot of them, or most of them, depending on how well you stir, are going to dissolve, because water is a tremendous solvent. Water breaks things down. That's one of the reasons why water is so good at cleaning. You put it over a surface, and whatever dirt or dust or dirty stuff is on the counter can be dissolved and washed away by the water. Other substances, other things that are other liquids that are not water, that don't have water in them, would obviously not have the same degree of ability to dissolve things, and therefore wouldn't be as good a cleaning agent. A solution means a liquid that has something dissolved in it. Salt water, for example, is a solution. We may not think of it as a solution. Solution we think of sometimes as being, funny chemical properties. But salt water is a solution because it's water that has broken down atoms and broken down molecules in them. Broken down salt molecules. Instead of the sodium chloride being there, now it's a bunch of sodium ions floating around and a bunch of chlorine ions floating around, and obviously it tastes a lot different than it would be just plain old regular water. And this is kind of a review. Salt is made of sodium and chlorine. They're held together by an ionic bond. The positive sodium ion and the negative chlorine ion. When the salt's dropped into the water, the sodium is attracted to the negative oxygen molecules that are floating around, and the chlorine to the positive ones, and this is eventually how the salt ends up dissolving. Not every type of molecule actually dissolves in water. Water dissolves polar molecules. In other words, molecules that have charges at different ends. Basically, ionically bonded molecules tend to dissolve more than molecules that have covalent bonds. Because if you remember when we discussed bonding, ionic bonding, and just for review, if you remember, this was an example of an ionic bond where you had one atom gave away its electron to the other one. Well, now if you think about these two things being bonded together, on this side you've got positive territory because you have an electron lost, and on this side you've got negative territory. Now if you think about putting this in water, well, the positive oxygen molecules will interact with this area, which is negative, and the negative oxygen molecules will interact with this area, which is positive, and that will end up being dissolved very well in water. Substances that are not as polar, like the methane we saw before, things that don't have strong ionic bonds, those are less likely to be dissolved in water. So water doesn't dissolve molecules with nonpolar covalent bonds because they don't have poles with a charge. They don't have sides, they don't have ends that would naturally be attracted to the positive or negative oxygen molecules. So for example, fats and oils are hydrophobic, which means that they do not dissolve in water. Water has an effect on these things. If you've ever put a piece of fat or something, or cheese or whatever it is in water, you know that it has an effect and mixes it around a little bit, but it doesn't dissolve to the same way that something like salt would. And finally, for this chapter, I want to just really do some vocabulary words that are going to be important later on in the course when it comes to the chemistry involved in living things. Synthesis means the building of two substances by combining them. You have two smaller substances, put them together in one big substance, now you've got synthesis. You take two monosaccharides, which means two single sugar molecules, and combine them into a disaccharide, which is a double sugar molecule. Common sense. When you do so by taking out a water, when you build two things by basically removing a water molecule from them and that combines them, that's called dehydration synthesis. In a chemistry course, we'd spend a lot more time on that. There's just an example: sodium chloride is formed by dehydration synthesis. This formation of table salt, I just made this slide. The reason why it's called dehydration synthesis is the way table salt is actually formed. You don't actually take the sodium and chlorine and put them together directly. What you need is, first you need NaOH, which is a sodium, an oxygen, and a hydrogen. That's called sodium hydroxide, plus HCl, that should be small l, which is a hydrogen and a chlorine, also known as hydrochloric acid. And what you get is an H2O, which is a water, plus a sodium chloride, which is a table salt molecule. That's how table salt is actually formed in a reaction. And it's called dehydration synthesis because what you're doing is you're making the sodium chloride by taking out a water, by producing a water. So it's called dehydration synthesis. The opposite is called if you look at the slide over here, a hydrogen ion is removed, and a hydroxyl, which is an O and an H, is combined into a water, and whatever's left over. So, if you look at this formula, what's forming the water? Well, this OH, which is called a hydroxyl, an OH, and this hydrogen ion, this H, and they're combined to form the water, and what's left over is this Na, that's sodium, and this Cl, that chlorine, and they eventually combine. That's why it's called dehydration synthesis. Hydrolysis is the opposite. If you imagine this is being reversed, you have the NaCl plus the I can write this just as easily down here. H2O plus the NaCl, and you add those two together to get sodium hydroxide and hydrogen and hydrochloric acid. You can just say reverse. Okay, well, I should probably spell that correctly. Make it a little bit bigger to make it a little bit more visible. There we go. Okay, so this is dehydration synthesis. You're producing a water. In other words, taking a water out of this stuff in order to produce sodium chloride. And the reverse is called hydrolysis. Basically, you're taking NaCl, adding a water, so that's why hydro, adding a water, and you're getting sodium hydroxide and hydrochloric acid. Just the exact opposite. One of the things we do when we digest simple sugars and various other types of molecules, we use hydrolysis to break it down so that our individual cells can use it. Here's just another example, very quick, basic example for hydrolysis or dehydration synthesis. This doesn't even really mean that much. All it is just an idea of sometimes you add water in order to break it down, or you can subtract water, draw out a water, produce a water, in order to link up two compounds to make one final product. So just a little review for the end of this chapter. Let's look at these two multiple choice questions, sample multiple choice questions. And we've got sweating is a useful cooling mechanism because water A takes up a great deal of heat in changing from solid to liquid. Can exist in two states at temperatures common on Earth, is an outstanding solvent, takes up a great deal of heat changing from liquid to gas, or ionizes readily. Well, many of these statements are true statements, but only one of them actually help. Remember, when you sweat, what happens is the hot molecules become vapor and move into the environment, while the rest kind of stay on your skin, and those are the cool ones. A, which is from solid to liquid, that's the melting. That's melting from ice to water. That has nothing to do with sweating. There's no ice involved in sweating. Can exist in two states at temperature common on the Earth. That's true. I mean, it happens to be true that there is solid water and liquid water at various places on the Earth, but that has nothing to do with sweating again. Is an outstanding solvent. Another true statement, but again, has nothing to do with sweating. What about D? Takes up a great deal of heat to change from liquid to gas, to evaporate. Yeah. Takes up a lot of the heat on your skin goes to vaporizing some of the molecules into the air, and the rest are the cool ones. And it ionizes readily, which isn't even a true statement. Doesn't even really make sense in the context of this question. So the only answer that really makes any sense is D. Number 2, you drop a handful of salt into a glass of water, which best describes what's happening. Okay, A, the positively charged hydrogen ends of the water molecule are attached to the chloride ions. Well, remember, chlorine tends to be a negative ion, because chlorine tends to gain an electron, and so therefore it would be attracted to something that is positively charged. So that makes sense. B, the positively charged hydrogen ends of the water are attracted to sodium ions. Now sodiums tend to be positively charged, so positive and positive would not attract to each other. So sodium and chlorine ions form a covalent bond. It's not true. They form an ionic bond. In any case, it has nothing to do with dissolving. And water and sodium form a covalent bond. That doesn't make any sense anyway. So the only answer that makes any sense over here would be A. A and B are both look like good choices, but you remember that sodium tends to turn into a positive ion, and chlorine tends to turn into a negative ion. So chlorine would be attracted to something positively charged, whereas sodium would be attracted to something negatively charged. Now we're going to kind of segue from our discussion of chemistry to molecules and atoms and things like that that are most relevant specifically to life. If this were a chemistry course, we'd look at the chemical makeup of other things like metals and things like that, but now we're going to focus specifically on things that are relevant to biological and relevant to life. Now, there are two types of biological molecules, in other words, two types of molecules that make up living things and the things inside of us, and those are organic and inorganic. Organic molecules are really loosely defined, they're molecules that contain a carbon and a hydrogen in them. There are some other scientific factors that determine what is organic, but for our purposes, for loose purposes, an organic molecule has a carbon and a hydrogen. An inorganic molecule has either only a carbon or only a hydrogen, or neither a carbon nor a hydrogen. Organic molecules also have carbon backbones bonded to hydrogen atoms. And one real functional difference is that organic molecules are capable of giving us energy, like sugar molecules, fat molecules, protein molecules are all organic. Things that are inorganic, like water, salt, carbon dioxide, may be necessary for us or for other forms of life, but they do not actually give us energy. Now, in terms of organic molecules, there are many different types of organic molecules. There are three basic types of nutrients that we eat that are organic. There are sugars, or starches, which are saccharides. Saccharin or saccharide comes from the same root of the word that means sugar. So starches are called saccharides. There are monosaccharides, which are simple sugars like glucose, fructose, sucrose, things like that. The basic structure of a monosaccharide is a single sugar molecule with 6 carbons. You'll notice there are 6 carbons along the outside of this hexagon. A C, C, C. That should be a C over there. It looks like an O, but it should be a C. Another C, another C, another C. Then you've got O's and H's kind of scattered around all over the place. There are some types of sugars with 5 carbons, but for the most part, sugars that we eat, like glucose and sucrose and fructose, have 6 carbons apiece. These are simple sugars. They can't be broken down because they don't need to be broken down. They're capable of being used by our individual cells for energy. In fact, more complex sugars, what we sometimes known as starches, get broken down into monosaccharides that our individual cells can use during the process of digestion. So polysaccharides, poly means multiple, so polysaccharides means multiple sugars, are large complex sugars, and they are really where starches, like breads, for example, are polysaccharides. So polysaccharides can be broken down into and used, for example, glucose molecules are stored in the liver as the polysaccharide glycogen. For when you have lots of extra sugars that your body doesn't need, your body converts the sugars into glycogen, which is, I guess, kind of fat, and the body stores. And then later on, when it needs it, the body takes it and breaks it down into monosaccharides that the body can use for energy. So this is an example, by the way, of a polysaccharide. A polysaccharide is the same kind of thing. You notice they're all the same basic sugar molecules, the same hexagons. You don't see the C's all over the place, but they are there. They're the same hexagons with a carbon, 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 and lots of hydrogens and oxygens scattered all over the place that are not in the document, but they are linked to each other. And then when the body needs to separate them, it uses hydrolysis to kind of break them apart into individual monosaccharides that the body can use. And so your body stores the extra starches or extra sugars as glycogen or some other polysaccharide, and then when necessary, when your blood sugar level falls and your blood needs more sugar to produce energy, to respirate in order to produce energy for your cells, the body takes some of that, breaks it apart into individual glucose molecules, and then goes ahead and uses them. Here are some examples of different types, or we're going to continue with the examples of different types of organic molecules. The previous slide was the sugar group. Now we're going to move to the fat group. Really, the three major groups are sugars, fats, and proteins. Fats there are saturated fat and unsaturated fat. Saturated fat, all fat is made out of fatty acids, which are basically carbon and hydrogen. And these fatty acids can be saturated, that are packed as closely together and as tightly together as possible. And they also can be unsaturated, which are not fully saturated with hydrogen bonds. And we'll see what that looks like in a minute. Fats, by the way, are also known as lipids. If you see that term, lipids and fats are essentially the same thing, fat molecules. Saturated fats, which is a diagram on the left side, notice all of the bonds are full with a hydrogen. All of the carbons have a hydrogen attached to it. You got a long row of carbons over here, and you got hydrogens attached with a single bond to every single carbon that exists. That's saturated. There's no other extra room for the unsaturated ones, where not every carbon is filled with a hydrogen bond. You can see over here there's a double bond between these two carbons, but neither of these carbons has a hydrogen. Over here, every molecule can have 4 bonds. Every carbon molecule can have 4 bonds, one in each direction over here. And here, the bonds are all filled. The bonds are all saturated. Each carbon's got 4 different things attaching to it. Most of these carbons in the middle have 1 carbon on each side and 1 hydrogen on top and on the bottom. Over here, on the other hand, these 2 carbons have no hydrogens. There are no hydrogens over here in this area. So instead, they've got double bonds to each other. They've still got 4 bonds, but instead of having bonds to each of 4 different other atoms, they've got they're double bonded with each other, and that's why it's called unsaturated, because they don't have the maximum number of hydrogens that are possible, and also there's a bend in the bonding causing an inability for the fats to get closely packed together. A trans fat, another type of fat, is a fat that's created artificially when food manufacturers turn liquid oils into solid fats by adding hydrogen or vegetable oils. One difference, by the way, is that saturated fats tend to be solid at room temperature. Saturated fats include things like butter and the congealed gravy that when you have gravy, or sometimes when you leave it out or put it in the refrigerator, it turns to solid. Chicken fat that turns to solid in the refrigerator, that's a saturated fat. Fats that are liquid at room temperature tend to be unsaturated fats, like oils, things like that. When food manufacturers, for various reasons, add hydrogen in order to turn what otherwise would be a an unsaturated fat into a saturated fat. That's called a trans fat. The process of making trans fat is called hydrogenation. And of course, you can probably guess the reason for that. It's because you're adding hydrogen. You add hydrogen to what ordinarily would be an unsaturated fat to turn it into a saturated fat. Food containing trans fat must be labeled appropriately. The general principle is that saturated fat is not good for you, and unsaturated fat, well, too much can still make you fat, but generally speaking, unsaturated fat is relatively healthy. It raises your good cholesterol, your HDL, whereas saturated fat raises your bad cholesterol, which is LDL. And so if you have to eat fat, it's definitely a good idea to choose the unsaturated fat variety that occurs in vegetable oils, peanuts, peanut oil, things like that, rather than saturated fat, which comes from things like gravy and butter and things like that. Trans fats are also not good, because trans fats, I guess theoretically they would be if they had remained unsaturated fats, but because they're turned into saturated fats, that's not good. So if you look at labels on food, government regulations now require food manufacturers to be forthcoming. I mean, this is just one example. Honestly, I don't even know what food this is, but you can see this 1 cup of whatever it might be. I don't know, maybe it's some kind of a pasta product or a drink or something, whatever it is. You can see this particular food has 260 calories per cup, 120 of them are from fat, 13 grams of fat. Fat, by the way, tends to have about 9 calories per gram, whereas sugars and proteins tend to only have about 4 calories per gram. That's why they say fat tends to make you fat, because, fat has a lot of calories per gram. So it's got 13 grams, about 120 calories, that's about right. Saturated fat, 5 grams out of the 13. So if you infer from this, there are 2 types of fat, saturated and unsaturated. There's also trans fat, which was unsaturated until the manufacturer made it into saturated fat. So there are 5 grams of saturated fat and 0 grams of trans fat, which of course means that there are 8 grams of unsaturated fat, which makes it a relatively healthy food as fat goes. And finally, our last type of organic molecule are proteins. Proteins are, not only do they give us energy, they also are a necessary component of many of the different things that we need in our body in order to function properly. The structure of a protein is a polymer of amino acids joined by peptide bonds. A polymer means a whole bunch of molecules next to each other. Amino acids are the building blocks of protein, just like sugar is the building blocks of starches. Lipids, or individual fat molecules, are the building block of fats. And so also amino acids are the building blocks of proteins, and peptide bonds just means the type of bonds that you see in protein. Now, proteins are fairly complex molecules. Not only do they have the sugar, hydrogen, and oxygen that, the carbon, hydrogen, and oxygen that sugars do, they also have an element of nitrogen in them. Here's an example of a peptide bond. Don't worry about the termini over here, but you've got 2 protein molecules over here. You see each of them have carbon, oxygen, hydrogen, and nitrogen, and this one also does, and the bonds that bring them together are sometimes known as peptide bonds. Many examples of important proteins that we have in our body include enzymes. Enzymes are found in cells, and they help with chemical reactions. They help do things in the body. For example, when the cell needs to break down a type of molecule, or when the cell needs to create or just do pretty much anything with it. The enzymes are little protein molecules that are floating around, and they're actually allowing things to break apart and be put back together and do all the little things that cells need to do on a daily basis in order for us to survive. You've got keratin, which is also a protein that forms hair, nails, scales, feathers, horns, other things that occur on the exterior of many different type of living beings. There are hormones. Hormones are secretions, liquids very often, that are necessary for different parts of the body to do different things. Insulin, growth hormone, adrenaline, glucagon, which breaks apart glucose. Things also like estrogen and testosterone, estrogen which is in women and testosterone which is in men, or really males and females, not necessarily men and women, are also examples of hormones. There are also antibodies. Antibodies, which are substances in the bloodstream, really white blood cells, that fight off infection, and they are made out of proteins as well. Toxins, things that could be poisonous, protein molecules can also be poisonous. We may not think of them as being necessary for life, but they may be necessary for animals to poison their pursuers, like snakes, which use toxins to defend themselves. Protein can also be a nutrient in food. It also gives us calories. It also gives us energy, as we discussed before, at the rate of approximately 4 calories per gram. Okay, and let's take a look at some end of chapter multiple choice over here. New government regulations require that foods containing trans fats be labeled appropriately, as we discussed. A trans fat is formed when manufacturers turn liquid oils into solids by adding hydrogen. This hydrogen process produces a solid fat. Why? Because, well, let's see. A. Allow fats to form tertiary and quaternary structures. Huh? Well, tertiary means triple and quaternary means quadruple or 4, but I don't know what that has to do with anything, so that is incorrect. Causes a phospholipid to form. A phospholipid would be something with a lipid and a phosphorus in it, but again, that doesn't really have anything to do with hydrogenated or solid or trans fats or anything like that. Allows fatty acid chains to pack together more tightly. Yeah, that's saturated. Saturated means when the bonds are totally packed together, so they are packed together more carefully. So certainly that looks like a good answer. Forms a wax molecule. Again, I have no idea what that has to do with anything. So the only answer, really, this is kind of an easy question. The only answer I see over here that makes any sense is C. Let's take a look at the next question. Sam tells Joe that organic, and by the way, when I'm doing these, what you can do if you like, I think I would recommend it actually, is to pause it and see if you know the answers, and then you can turn it back on and see about my explanation. Sam tells Joe that organic molecules are made out of carbon backbones with hydrogens. Okay. Joe doesn't understand how there can be so many different organic molecules if they're all made up of the same basic components. Sam explains that organic molecules, okay, so we're looking for something here different from organic than inorganic. A, vary because they possess different isotopes of carbon. Well, we didn't even discuss what isotopes are, but that means different types of carbon, but that has nothing to do with it. Well, organic and inorganic can contain the same kind of carbon. B, actually all have the same number but different structure, but different numbers of electrons. Not necessarily. That has to do with bonding, not different types of organic molecules, which are really based on the different numbers and types of bonds and things like that. C, are different because of the different types of hydrogen bonds that forms. Well, that actually could be true. It's not a terrible answer. It could be true in certain types of, for example, a difference between a saturated fat and an unsaturated fat, but that's only a very narrow distinction. In the obviously the types, whether they have different types of hydrogen bonds is not going to be the difference between a protein and a sugar, only between a saturated and unsaturated fat. So C may be an answer for part of it, but D looks like the best answer because they vary based on different functional groups. They have different, some function because they've got combinations of carbon, hydrogen, and oxygen, whereas proteins have nitrogen also, etc. So D looks to me like it is the best answer in this case. Next up is a discussion of cells, the foundation of life, the basic building block of every living thing are cells. Cells are individual. Human beings have millions and billions of cells. There are some types of organisms that only have a single cell, like for example a bacterium. A bacterium, which make up bacteria, which is the plural of bacterium, contains only a single cell. But every living thing is fundamentally made up of cells. The two basic types of cells are prokaryotes and eukaryotes. Prokaryotes are cells that are undeveloped. Eukaryotes are cells that are more developed. Basically, very, very basic organisms like bacteria are prokaryotes, and eukaryotes are things like animal and plant cells. Now, the main difference is that eukaryotes, the bottom one, the more advanced one, have a defined nucleus in the middle. A nucleus is the thing that has the DNA, and the DNA is the thing that really runs the cell. We're going to look at some examples as we move along in the chapter, but this is an example of how a cell generally looks. You've got the cell membrane all the way on the outside. We're going to look at the various organelles, which are these things in the middle. But in the very middle over here, you have what's called the nucleus. And the nucleus is where you have the DNA. On the other hand, when you're talking about a prokaryote, which means before, pro always means before, you've got something like this, a prokaryote cell. It's similar in terms of the basic structure. This one happens to have a cell wall, but that doesn't matter. What does matter is there's no defined nucleus. The DNA, this orange stuff in the middle over here, isn't, whereas before we saw the DNA would be somewhere over here in this area, that would have a defined nucleus in the middle. A prokaryote cell over here would not have a defined nucleus. Instead, the DNA would just kind of be swimming around in the middle of the cell. Okay, so the prokaryotes are in bacteria, things that are very, very not advanced, things that are very primitive. Whereas eukaryotes are things like plant and animal cells. They have a real nucleus, and we're going to discuss a little bit later on the distinction between the plant cell and the animal cell, but certainly there are many very important distinctions between them. Now there's basically a distinction between animal cells and plant cells that we'll look at in a little bit, but first I want to look at the various things that are inside a cell. A cell's got lots of organelles in the middle. This is just an example of, well, this happens to be an animal cell. But you can see in this one, and in the one that we looked at a little while ago, that there are many different materials inside the cell, and those are called organelles, kind of little organs. You've got the lysosomes. The lysosomes contain the digestive enzymes. In other words, they break down food. They break down polysaccharides into simple sugars, big amino acids into individual proteins, big fat chains into individual lipids, the sort of thing that are necessary for the cell to break down whatever nutrients are necessary. And they also break down defective organelles. If some of the mitochondria or whatever it is aren't working, they can be broken down and recycled, or at least gotten rid of through the waste process in the body. You've got the membrane. The membrane is the outside of the cell, kind of the thing that stops materials that are supposed to go in, or that are supposed to stay in from leaving and supposed to stay out from coming in. It's made of phospholipids, which are fats with phosphorus. That's why it's called phospholipids. Normally, lipids only contain carbon and hydrogen, but there are phosphorus molecules involved as well in the plasma, in the membrane, and proteins in the membrane as well. The job of the proteins is to communicate, really to facilitate the communication between the cell and the environment. In English, without going through, you can read this on your own, obviously. But basically, what the membrane does is specially structured and specially engineered, not just to keep things in or out, but also to allow transport one to another. Sometimes things have to leave the cells if the cell wants to get rid of it or transport it. And then there are some things that have to go into the cells if the cell needs the materials, sugars, proteins, whatever the cell needs. Cell membranes look something like this. You see, you've got the lipids, you've got picture this as being inside the cell, and up here as being outside the cell. And you've got this structure, the main structure all over here, are fat cells that are blocking things from coming in and blocking out and coming out. But you've also got protein molecules, protein channels that kind of open and close to allow things necessary to move in and move out. And those are the things that actually control ingress and egress from the cell, which means moving in and out of the cell. That's, of course, the function of the membrane. Some other things, we've got vacuoles and vesicles, which transport and store also materials. If there's water or food that the cell needs and is going to keep for a later period of time, that is what is known as a vacuole or a vesicle. The ribosomes produce the protein for the cell. take the amino acids that are imported by eating or whatever other process the cell gets it, and then go ahead and produce whatever proteins are necessary for the cell. We learned last time all about the different types of proteins that you have, things like hormones and enzymes and materials that move information in and out of the nucleus. These are all things that are necessary that are produced by the ribosomes. You have the endoplasmic reticulum, or the endoplasmic reticulum, excuse me, the ER, which synthesizes membrane components and allows things to travel throughout the cell. We're going to look at a diagram in a minute. The Golgi apparatus, or the Golgi bodies, packages and sorts proteins and lipids, kind of the manufacturing plant, or really the packaging plant. The manufacturing plant would be the ribosomes, and then the Golgi apparatus would be the packaging plant, where it puts everything together and moves it around through the endoplasmic reticulum in terms of where it has to go. Other things that could be involved in certain cells, you've got cilia. Cilia are kind of like hair-like structures, and flagella are longer, whip-like structures that allow the cell to move around. The cilia kind of allows the cell to feel things, the flagella to actually move things. Some certain types of bacteria or certain types of protozoans, which are tiny, tiny, tiny little organisms that live in water, including in our bodies and in our mouths and whatnot, travel around through the flagella. The nucleus contains the DNA, contains the genetic material in the inside. The chromosomes are they make up the DNA. DNA is made up of chromosomes. The chromosomes are the thing that essentially tells our body what to be. The reason why you are a human being and not a fruit fly, I mean, aside from everything else, is because of the fact that your chromosomes indicated that you should grow a brain and grow eyes and grow whatever, and the fruit fly only indicated that it should grow a small little set of wings and whatnot. That doesn't mean that you're in danger of turning into a fruit fly if your DNA changes a little bit. Our DNA is quite a bit different from the fruit fly's DNA, but the fact of the matter is that the chromosomes are kind of the blueprints on which the cells know what to do, and the cells go out and do their jobs because those jobs are written in the blueprints. The nucleolus is the kind of the protein synthesis hub inside of the nucleus. The nucleus itself in the middle has the nucleolus in the middle, which is where the proteins are generated. Essentially, the nucleolus, it's kind of complex. I don't want to go into it right now, but the chromosomes, namely the DNA, have to be able to send its message throughout the cell, and the materials by which that message is sent are produced in the nucleolus. The nuclear envelope covers the nucleus, just like the cell membrane covers the cell itself. You have the mitochondria. The mitochondria does the energy production, the respiration. The mitochondria is the thing that takes the sugars that we eat or that we have and generates energy. It does it by a fairly complex process called respiration, either by aerobic respiration, anaerobic respiration. I don't want to get into that right now. But the mitochondria are the thing that create something called ATPs, adenosine triphosphates, which essentially are able to transport energy. It's able to store energy in these ATPs. The ATPs can go wherever they're needed and release the energy that are necessary. Then you have the centrioles. The centrioles are necessary as part of the reproductive process when one cell splits apart into two cells in a process called mitosis. And then you have the cytoskeleton, which really just forms the more solid parts of the cell. Things like flagella, cilia, things like that. These are all different, and this is not even an exhaustive list. There are plenty of other things that may appear in an individual cell. And like it says over here, don't necessarily memorize the list, but you should be able to distinguish between different types of cells and the functions of what some of these things do. A plant cell is slightly different than an animal cell, in that, except that they have a cell wall in addition to a cell membrane. They also have big plastids they kind of look like very big vacuoles, very big storage areas. They have chloroplasts, which are necessary for photosynthesis. Photosynthesis means making food. A plant makes its own food, whereas a human being doesn't make its own food. I mean, it can cook its own food, but it doesn't actually spontaneously generate its own food. It has a central vacuole, a much bigger vacuole than an animal cells. Plant cells don't have lysosomes. Flagella and cilia are very often involved in movement, and plant cells, of course, don't move, so they don't need that sort of thing. Here's an example of a side-by-side comparison. I have some diagrams that'll be a little bit easier to show you, but just as a general principle, you can see some of the distinctions. The plant cell's got a cell wall, or the animal cell doesn't. The animal cell is more circular. The plant cell is more square. The plant cell's got a much bigger vacuole over here in the middle that stores a lot of food and stores a lot of water and whatnot. But they both have nuclei, they both have mitochondria, they both have a lot of the other things that are necessary. Here are some other examples. Here is an animal cell fairly round, and it has a nucleus in the middle with the nucleolus in the middle of the nucleus, lysosomes, and a plasmic reticulum, which is kind of the transport mechanism from the nucleus to other parts of the cell, the Golgi apparatus, the mitochondria, which remember do the respiration for the cell. Then here's an example of a plant cell. Plant cell looks again fairly similar. You have the cell wall outside the cell membrane. You've got the Golgi apparatuses, just like I guess, just like you do in the animal cell. You've got chloroplasts. These do the photosynthesis, which you do not have in the animal cell. You've got the nucleus and the nucleolus in the middle over here. You've also got a huge vacuole that takes up a lot of the cell. The last half of this chapter goes more in depth into the cell membrane and materials moving from inside to outside and from outside to inside of the cell. Every cell, whether a plant or an animal cell, has a cell membrane, which is an outer layer that is surrounding the cell. Its main function is to keep things in that are supposed to be in and keep things out that are supposed to be out, but also, of course, to allow things in that are supposed to come in and to allow things out that are supposed to come out. They regulate biochemical reactions by allowing the ingredients for those biochemical reactions in and out of the cell. They separate the organelles from the surrounding interstitial fluid, which means the fluid that all the cells are kind of bathed in. You know, all the cells in the body, let's say in particular in the skin or in your heart or whatever it is, are kind of bathed in this fluid that where all the cells sit in. Well, you want to stop or prevent that material from interfering with the organelles. It also separates between the membrane that are enclosed within the organelles within the cell and the material outside the cell. It also creates an attachment in between cells by latching onto other cells, hooking up with other cells, moving next to other cells, and allowing things to be moving back and forth. That's really the last thing, to regulate the exchange of substances, to make sure that the material inside the cell can get out, and the material outside the cell can get in. And a lot of the movement between inside the cell and outside the cell, and between different cells, is based on the materials being dissolved in water, and the water moving from inside the cell to outside the cell. Obviously, it's a lot easier for the cell to move water in and out, because water is very, very small and very permeable. It moves into materials a lot easier than solid materials. Now, that doesn't mean that solid materials can't move from outside the cell to inside the cell. They can, but it's a lot easier when they're dissolved in water. A substance that can be dissolved, for example like salt, is a solute, whereas the liquid in which it dissolves, in this example of course water, is the solvent. So when you have salt water, the salt is the solute and the water is the solvent. Concentration is the amount of solute that is dissolved in the solvent. You could have salt water that is very concentrated, or you could have salt water that is not concentrated. Salt water could be a little salty, or salt water can be extremely salty. So concentration of the solute is a measure of volume. For example, just a very simple example, if you have 2 cups of water and you've got 5 tablespoons of sugar in one and 3 tablespoons of sugar dissolved in the other, obviously the first cup has a higher concentration than the second. If you were measuring the amount of material that is dissolved, the first cup you would say 5 tablespoons per cup, and the second one you would say 3 tablespoons per cup. Very simple. Obviously that's a 5-to-3 ratio, and the first one has the higher volume. Now, the thing about materials moving inside the cell and outside the cell is that water is always drawn towards an area of higher concentration. Salt draws the water towards it. So that's why, for example, when you salt, if you want to get rid of the blood in a piece of chicken, you can salt it. You can put the salt in on it, and the salt kind of draws the water out of it. That's why salting a chicken works to get rid of the blood. And salting anything works to draw the liquid out of it. Water is drawn from area to an area where it's a higher concentration. So, for example, if you have salt water outside the cell and non-salt water, fresh water inside the cell, the water from the fresh water is going to be drawn into the outside. It's going to be drawn to the area of where there's more concentration, of where there's more salt on the outside. Materials themselves, like the salt itself, tends to move from areas of high concentration to areas of low concentration. If you, for example, have very heavily diluted salt water, very salty salt water, and a very not diluted salt water, very not salty, watery salt water, and you pour from one to the other, of course the salt is kind of going to spread out and even itself out so that eventually the entire thing is going to be pretty uniform. The entire cup is going to be pretty much the same. And generally speaking, whenever you have something dissolved in water, the material, the sugar, the salt, the solute, whatever is dissolved in it, is going to move from an area of high concentration, where there's a lot of it, to an area of low concentration. That's why eventually everything is going to even out. That's a very important concept in the concept of things moving into and out of a cell, because whenever you have a higher concentration outside the cell, whatever it is in higher concentration, it's going to tend to try to move through the cell membrane into the cell. Gradient is how you measure the difference in certain properties. If you have highly concentrated inside and lower concentrated on the outside, there is a gradient, which means a distinction between the two. And that example we discussed before, when you had 2 cups on one, 5 tablespoons per cup on one side and 3 tablespoons cup on the other side. So there's a drop-off if you move in between those two regions, if you move in between those 2 cups, you're going to have a drop-off in concentration from 5 to 3. That's called gradient. Gradient is also sometimes used when you're talking about, hills, a distance in elevation. A gradient, if you have a steep hill, you're going from a higher elevation to a lower elevation. That's also called a gradient. So in this context, it means a difference in concentration. Now, a cell membrane is selectively permeable. Permeable means something can infiltrate it, something can move into it and out of it, and selectively means only some things. Not everything can move in and out of the cell membrane, but on the other hand, it's not completely airtight or watertight, which means some things can move into and out. All those concepts really brings on our next issue, and that's the issue of how things get transported into and out of the cell, and whether energy is necessary for that process. The 2 types of transport of which materials can move into and out of a cell are passive transport and active transport. Passive transport does not require energy, whereas active transport does require energy. And to visualize this, I know it may seem very complex in the abstract. I want you to visualize, instead of the cell membrane being a cell membrane, visualize it being a paper towel. And you've got liquid on the outside of the paper towel, and you've got liquid on the inside of the paper towel. Now, yes, if it were a paper towel, it would tear fairly easily, but imagine it's the kind of paper towel that doesn't tear. Maybe it's Bounty, according to their Bounty commercials. So we're going to think about it that way in terms of something being selectively permeable, where not everything can move in and out, but some material can move in and out of beyond the paper towel. Passive transport is the sort of thing that happens automatically. either because the concentration is lower inside than it is outside, or because it's water which can just kind of slide through the walls. Whereas active transport, where you have a big molecule that cannot so easily go between the inside of the cell and outside of the cell, it requires energy in order to move it from one place to another, and that is what you would call active transport. So let's take a look at what would constitute passive transport. Passive transport is also sometimes known as diffusion. Okay, let me have the wrong slide up, and now I'll put the correct slide up. Anyway, so diffusion of substances happens across a membrane down a gradient of concentration, pressure, or electrical charge, and don't require energy. Diffusion, or passive transport, occurs either when you're going from a region of high concentration to low concentration, like the salt moving from salty salt water to non-salty salt water, or to regular water. That happens automatically. It happens naturally, because things tend to move from areas of high concentration, where there's a lot of it, to areas of low concentration, where there's not that much to it. So that does not require energy. Pressure. Things tend to move from areas of high pressure to areas of low pressure, just like wind. Wind is where you have high air pressure in one area and low air pressure in the other, and the high air pressure blows to the area of low pressure. And electrical charge, which we'll get to in a little while. Electrical charge can also naturally allow things to move from one area to another. The types of passive transport that we're going to look at are simple diffusion, which is a diffusion of either water, which naturally moves from place to place, like through a paper towel, lipid-soluble molecules, or gases, dissolved through the membrane. In other words, things that are so small or so dissolved in the water that they can kind of squeeze in between these fatty materials over here. As you can see, these fatty materials on the outside of the membrane, if you have water over here, it just kind of squeezes through the little holes and goes inside the cell. Or if you have material that is concentrated, or a material that is dissolved in the water, it can just kind of move through without any help from these big red things that allow certain things in and out. That's one. The other type is facilitated diffusion, which is water or water-soluble molecules through a membrane via a channel or carrier protein. That's something that goes through these red proteins over here, transmembrane proteins, where the proteins kind of, as we looked at last time, kind of open up a little bit and allow things that wouldn't ordinarily be able to pass through these fatty substances, either because the materials are too big, they're not dissolved enough, and they need the membrane to kind of open up, and that's what these proteins do to allow them in. They still could be passive transport, because remember, if they're coming from an area of high concentration up here to an area of low concentration down here, or high pressure up here to low pressure down there, they still don't require energy, but they still need something to open up and allow them in, and that's the purpose of these membranes. And then we have osmosis, also a form of diffusion, also a form of passive transport, which is diffusion of water across a selectively permeable membrane, like the ones on the outside of cell, from an area of higher free water concentration to an area of lower free water concentration. In other words, there's room for more water to come in, and that is what is sometimes known as osmosis. A couple of other things about passive transport, about diffusion. First of all, it occurs when the temperature is higher. When particles of sugar are moving from place to place, again, the example is pretty obvious, it'll diffuse faster at a higher temperature at 75 Celsius than only at 45 degrees Celsius. In general, things that are hotter, environments that are hotter, are more conducive to dissolving things, are more conducive to chemical reactions in various different ways, in many, many different contexts. One example you might realize is that from your own experience, you probably know that things tend to dissolve faster in hotter liquid than in colder liquid. If you're putting sugar in your coffee, if your coffee is cold, it's going to take a lot of stirring, and even so, you might not get all of the sugar to dissolve. On the other hand, if your coffee is piping hot and your coffee is boiling and you put sugar in, it's going to dissolve pretty much 100% and pretty much right away. The greater the concentration gradient, the faster diffusion will occur. In other words, the more the concentration difference, the faster things will move. If you have very very salty salt water and you put it next to you mix it in with non-salty salt water, you put it in, like, let's say, a paper towel in between, it'll move very quickly. Whereas if the concentrations are pretty close, it may not move as quickly from one side to another. Things tend to even out as quickly as possible, so the higher the gradient, which means the higher the difference in concentration between the two sides, the faster one will move to the other. Example, you've got particles of sugar being diffused into two cells. Inside the first cell, the concentration is 25, and the second is 35. The gradient is relatively light. Well, forget the gradient between the two. Both cells are in a solution that has a 60% concentration. So the gradient between the 60 and the first cell is 60 to 25, whereas the gradient between the solution and the second cell is 60 to 35, which means the sugar water, the one with the sugar will move from the solution, which has the higher concentration, to the cells, but it will move faster to the 25% cell because the gradient is more, the difference in concentration is more, than the other one, which is the 35% cell. Osmosis works very similarly. Remember, osmosis is really just a form of diffusion when you're talking about water and water with a concentration of materials with a solution where the materials are dissolved in the water. And so this is, again, where you have water across a selectively permeable membrane, like a cell membrane, from an area where there's more water to an area where there's less water. So when and again, all of these things that we're going to discuss now are really just corollaries to what we discussed. There shouldn't be any major surprises over the next couple of slides. The more solute a solution has, namely the more concentrated it is, the less free water there is. Therefore, there's going to be, the more empty the water is, the faster the water with solution, the faster the water with dissolved particles will move towards it. Hypertonic means a solution with a greater concentration than the solute, and hypotonic is the opposite. Hypo means less, hyper means more, generally speaking. And so hypotonic means with more, it's just a terminology. Hypotonic is a solution that is more diluted, which means it is less concentrated. So just to give you an example, if you have a hypertonic solution next to a hypotonic solution, that means you have a more concentrated solution next to a less concentrated solution. The solutes, the sugar, the salt, whatever is dissolved, will move from the hypertonic solution to the hypotonic solution. Again, just to reiterate, hypo means less, hyper means more. Hyperthermia is when you're too hot. Hypothermia is when you're too cold. Isotonic is when the two things are equal. You have two solutions that are next to each other, separated by this membrane, by this paper towel, whatever it is, and they have either concentrate. Can you guess about what's going to happen in terms of movement? You can pause it and think about it. Okay, now that you've unpaused it, the answer, of course, is that there is not going to be much movement between one and the other because their concentration is equal. So water moves by osmosis from the hypertonic solution, which means the more concentrated solution, to the hypotonic solution, which is the less concentration solution. Here's an example, again, extremely obvious. It allows water to pass, but it doesn't allow salt water to pass, but the cell contains a 3% salt solution. The cell is then placed in a 5%, so obviously water will move out in the cell because it is going to try to equalize the amount of the concentration overall. If, for example, the cell and the water outside the cell was the same amount of material, eventually there would be 4% concentration across the board. That's also why water tends to be drawn out of salt water, as we discussed earlier, because the salt is drawing the free water in order to equalize the concentration. Osmosis across a plasma membrane is absolutely critical for all living things. all plant cells, and actually animal cells as well, are supported by water that moves into the cell. A plant cell has that huge vacuole where it stores the water. How does it get the water? Well, obviously it has to get the water from outside the cell. And how does it do so? By diffusion or osmosis through the cell membrane. Plants that are not watered, plants that don't have enough water, well, the plant itself will shrivel up and shrink, and the reason is because the vacuoles are no longer filled with water, causing it to have the same structure that it normally would. Organisms living in fresh water have to make sure that they don't absorb things from the outside that are not good for the cell. They have to use energy to counteract osmosis because otherwise things that are living, other organisms, things that you don't want inside your cell, would automatically flow through the membrane into the cell. So it actually has to use these proteins in the outside to keep things out. Here's an example. A toad egg cell is placed in a hypotonic solution, which means in a solution with less concentrate. It will swell via osmosis. It will get bigger via osmosis because hypotonic again means there is less solution in the inside, which means the water will flow from the outside to the inside. And to the extent that that is not good for the cell will have to use energy in order to keep it out. Osmosis, of course, also helps with the transport of nutrients from inside the cell to outside the cell. An example, bacteria, this one, Vibrio chloride, it doesn't really matter what the bacteria is, but a particular bacteria releases a toxin that blocks a channel protein in the membranes of the cell, and that's one way it keeps sodium from moving from the intestine into cells. That's just one example of a particular type of bacteria that helps human beings, that lives in a human being or an animal digestive tract, and in this case prevents sodium ions from being transported into cells. Water, as we discussed before, leaves the cell and enters the intestines through osmosis. Now this little diagram serves to illustrate what happens to cells when they go into liquids that have different concentrations. Now the ideal thing for a cell is to have the same concentration as what's outside. That's the middle diagram. The middle diagram is isotonic, where you have the same concentration of what's inside the cell to the solution that's outside the cell. And so you got the same amount of water, you got water going in, you got water going out, you got the same amount of water going in and out of the cell, and so you have no problems. A hypotonic solution on the left, this is where you have a higher concentration inside the cell than outside the cell. Now when you have a higher concentration inside the cell than outside the cell, so the water, the solution, the materials, the salt, the sugar, whatever it is, would ordinarily go out of the cell. But if the cell membrane didn't let them out, as is the case with many materials that are dissolved, the water gets drawn inside the cell. Remember, overall the water, once the concentration is going to equalize between inside the cell and outside the cell. And so what happens there is the cell is eventually going to burst, because what's going to happen, the water is going to be drawn inside the cell. The hypotonic solution, which means the outside of the cell, has less concentration than inside. So the water is going to seek the area where there's more stuff, which is inside the cell, and the cell is going to burst. What you have over here, what you have on the right side, is the opposite. You have a hypertonic solution where you put a cell, let's say, for example, in salt water. Now, the salt water is a higher concentration than the cell, and therefore the water is going to be drawn from inside the cell to outside the cell in order to equalize the concentration, and the cell is kind of going to shrivel up. This is what happens when you salt chicken and the blood gets drawn out into the salt. But if you put that's why if you put cells like bacterial cells in salt water, they're probably going to die, because the water from inside is going to be drawn outside, and the thing is going to end up shriveling up until it has not enough water inside to function, and it's probably going to die. So now let's look at some end of lesson multiple choice questions over here. And first we have question A. What would happen if the plasma membrane of a human cell or a regular cell, an animal cell, doesn't matter, were composed solely of phospholipids, which means those fat cells, those orange fat cells on the outside, and no proteins, none of those things in the red things in the diagram that open up to allow things in and out. So it's a good question. What would happen? Would it all movement across the membrane cease? No, not necessarily, because remember, water can diffuse, water can move in between the little crevices of those phospholipids, of those fatty things. Water can kind of just diffuse in and out, so that's not true. Facilitated diffusion, active transport, and osmosis would not occur. Again, not true. First of all, osmosis could happen. Active transport that requires energy could certainly happen. The cell could kind of swallow. We didn't discuss it in this chapter, but the cell can swallow what it needs to from the outside through active transport, so that's certainly not true. C, the movement of molecules across the membrane would not be affected. That's also not true, because if you wouldn't have the proteins to open up, then the cells that need to get in and out would not have as easy a time doing so. Immune reactions would not be affected. We didn't really discuss that in this chapter, but that's also not true. White blood cells would certainly be affected by not when it attacks things or tries to swallow things up, the proteins are necessary for that. E, of course, is the only one left, and why is that true? Because simple diffusion and osmosis, water moving in and out of the cell, would definitely continue to occur even if there were no proteins, because that only requires the crevices between the lipids, which it has. So we're going to choose E for this one. A cell from a wheat plant, it's a plant cell, the fact that it's wheat makes no difference, it contains all of the following except except a what? A flagellum, a cell wall, chloroplast, mitochondrion, or a cytoskeleton. Well, a flagellum is something that causes the cell to move. That's like a little tail, a little whip tail that allows it to move. Does a wheat cell, a plant cell, move? The answer, of course, is no. So A is the correct answer. A cell wall, well, that a plant cell does have that. Plant cell also has chloroplasts. All cells have mitochondrion for the respiration, and all cells have cytoskeletons, which are inside the cell that make up the materials like the membrane and other materials that the cell needs in order to have its structure. So the best answer here would clearly be A. Next, we're going to discuss energy flow and in the life of a cell. Lesson 4. Energy flow, of course, is making sure that things can move from one point to another point through active transport and to allow other life processes to be done that need energy. Energy in general, and this really has nothing to do with biology, this slide, this is really more physics, but it's really very, very basic physics. And energy means the capacity or the ability to do work, which means to move an object, to move any object. When I pick up a pen and put it back down, I am doing work. Work requires some sort of energy. Energy can be mechanical energy, which we're not really going to focus on very much. Things like when you chew your food or when you smack something with your hand or anything that's mechanical energy. The energy when it comes to food and the energy that food gives you is really chemical energy, which is the ability to do work through manipulation of atoms, molecules, things like that. Chemical energy is available in the bonds of molecules. Really, the way your body works in terms of producing energy, it stores energy in ATPs, adenosine triphosphates. When there's no energy in it, it's an ADP, which is a diphosphate. Again, that's a little bit more complex than we're going to go into right now, but that's a form of chemical energy that your body uses. Energy can be potential, which means it is stored, which means you can harness it, you can use it. If I pick up a pen and I hold it 5 feet above the ground, it has potential energy because all I need to do is let it go and it will fall down, and I don't know if you heard that, but that was the pen falling on my desk. Kinetic energy is the energy of actual movement. Now, so when it comes to food, when it comes to biology, energy in food, the 100 calories in your piece of bread, that is potential energy. The kinetic energy is when the energy is actually being utilized by your body. So, for example, to pick up the piece of bread and put it into your mouth, that uses kinetic energy, and it also puts it into your body where your body is able to release the stored potential energy as kinetic energy. Kinetic energy can be turned into potential energy, and potential energy can be turned into kinetic energy, or whatever. They each can be turned into each other very simply. Just one quick example, picking up a pen, I'm using kinetic energy to pick it up, it now has potential energy. I let it go, the potential energy is now being converted back into kinetic energy as the pen falls. Another quick example, plummeting a roller coaster car is turning kinetic when the roller coaster goes up, 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 up. It's using kinetic energy. Now it's high above the ground. It's got potential energy, and then when it comes back down the other side, it is now using kinetic energy and losing the potential energy. Food, as we discussed, has potential energy, and photosynthesis is when the plant makes its own food, when the plant makes in its chloroplasts, creates sugar, and it creates sugar potential energy that the plant can later use using the kinetic energy of the sunlight. A couple of other rules, also in physics, that are relevant to biology in our case are the rules of thermodynamics. Energy cannot be created or destroyed by ordinary processes. It can only be converted from potential energy to kinetic energy, back to potential, back to kinetic, etc. You also, of course, when you use energy, when you run around, when you sweat, or whatever it is, you're causing an increase of the molecular movement. When you cause molecules to move faster, you're causing things to get hotter. In other words, the energy is just being transferred from place to place and from object to object. It's not actually being lost or gained. Nuclear reactions are a different issue, but I don't even want to get into that. That's not really an issue relevant to biology and life processes. In a closed system, where energy cannot enter and cannot leave, the total energy always stays the same. The total amount of energy out there always stays the same. It may be converted, it may be changed form. It used to be when you run around and you are using the potential energy originally existed in the food that you ate, and then it converted into kinetic energy when your body converted it into the energy necessary for you to run, and then that generated more heat, and that heat caused your sweat molecules to evaporate in the air, and then the air is becoming a little hotter because of your kinetic energy, and the energy. But there's always the energy there's always energy there. It's just a question of which form it takes. The second law of thermodynamics is that when energy is converted from one form to another, the amount of useful energy, in other words, the amount of energy that you can actually use and harness, decreases. It's related to the rule of entropy, which says that things tend to get spread apart and less organized as they get further apart. An example is a very simple example. When you exercise, you burn the food that you have in you, the sugar molecules, the fat molecules, those are potential energy. They have energy stored in them. Then, when you exercise, you use kinetic energy to burn the food or cause energy to be received from the food. And that energy that you produce is given off into the person's surroundings and makes the area around you a little bit hotter, as we discussed before. How do you get energy into the earth? The answer, of course, is basically sunlight. Because all the energy that we get in our systems ultimately comes from the sun. We eat other animals or other vegetables or whatever it is, but everything ultimately eats something that is a plant. And in all the bottom of each food chain, when we eat the cow ate the grass, is some sort of a plant, like the grass. And the grass has to make food, and the plant makes the food through photosynthesis, through its own processes, by converting sunlight. No energy conversion is 100% efficient, and that's why things tend to get less and less efficient as you move along, and things tend to get more and more random and more spread out. Now, something has to facilitate the conversion of energy from potential energy to kinetic energy, and that is sometimes known as activation energy. A quick and simple example of something that we can think of that changes potential energy to kinetic energy. You have a rock at the top of a hill, and you have to push the rock in order to start rolling it, and then it picks up its own momentum and goes the rest of the way down the hill. So, of course, because of the fact that the rock was on the top of the hill, that it had a lot of potential energy. When it was rolling down the hill, it was using that potential energy and using kinetic energy to actually roll down the hill using the force of gravity. But when you pushed it originally, in order to get it started, then you were activating it. You were using that's called activation energy. Activation energy overcomes the repulsive electrical forces between electron shells so that they can move close enough together to react. In English, what this means in chemistry, In order for two things to combine, let's say a sodium and a chlorine, the electrons have to join together. The sodium has to lose an electron and the chlorine has to gain an electron. I think, we discussed this a little bit earlier. Let's look at the diagram again. This is an example of a sodium chloride diagram. Remember, the sodium had 11 electrons originally, and the chlorine had 17. And the sodium gave away its electron, the yellow one, to the chlorine, which now has 18. Now, electrons in general are both negatively charged, and negatively charged forces repulse each other, just like positively charged forces repulse each other. Negatively charged forces also repulse each other. Now, so in order for this to happen, in order for this electron to move in and join with the chlorine's electrons, it had to overcome the repulsive force, because these electrons would normally, the green ones, would normally repulse the yellow one. So in order to have them join up, and in order to become a sodium chloride, you need to have energy to overcome that. So anytime you have two atoms that are combining with each other, it means their electrons essentially are going to combine, and that's talking about whether you're talking about an ionic bond or a covalent bond. This basic idea is the same. That is, you need some sort of energy to overcome that repulsive force so that they join. Now, that can come from many different sources, but in general, when you're talking about life processes and combinations, it requires some sort of energy. A chemical reaction is any kind of a reaction that changes the atomic structure. It could be sodium and chlorine joining. It could be sodium and chlorine splitting apart. It could be hydrogen and oxygen joining. It could be methanes, the carbon and the hydrogens of methane splitting apart. Anything to that effect. Anything that breaks the chemical bonds that hold the atoms together, or of course the opposites, establishes the chemical bonds that hold things together, is called a chemical reaction. Now, a chemical reaction can need energy, like when you're taking two things and stuffing them together, so taking a sodium and taking a chlorine and stuffing them together and overcoming the repulsion that the electrons would normally meet with each other, then you need energy in order to do that. You also, on the other hand, could have reactions that release energy. There was pent-up energy when the sodium and the chlorine come apart. Well, then the energy that's no longer needed to overcome the repulsion between the electrons gets released into the environment. And those two different types of chemical reactions are called exothermic reactions or endothermic reactions. Exothermic reactions are when energy is released, whereas when an endothermic reaction, the energy is required to put in. An exothermic reaction produces energy, usually in the form of light or heat. Could be in other electromagnetic forms, but the release of energy is usually in the form of light or heat. And an endothermic reaction requires energy to put it in. Exothermic reactions release energy. Endothermic reactions gain energy. Exothermic reactions tend to make the surrounding environment hotter, whereas endothermic reactions tend to make the surrounding environment cooler. So, for example, when you put a log in a fireplace, you're hoping, and you're probably going to get, an exothermic reaction, because energy is released. The reactant has more energy than the product. Some of the extra energy that's being used gets released into the environment. Sometimes you actually have both of these reactions that are happening at the same time, and the exothermic reaction produces energy that fuels, or that establishes the energy necessary for the endothermic reaction. That's what's called a coupled reaction. Two reactions that provide energy one for the other. The exothermic reaction, which gives off energy, which releases energy, gives the energy needed to drive the endothermic reaction. Uses ATP or electron carriers as intermediaries. I don't want to worry about electron carriers. That has nothing to do with biology. But again, I've mentioned it many times. At some point, we should actually look at what they are. ATPs, which has an adenosine or an adenine and 3 phosphates, and that's a carrier of energy. Long story short, what the body does is it takes the these ADPs, which has an adenine and two phosphates, and the mitochondria uses energy to take a third phosphate and scrunch it in there to make it an ATP, make it an adenosine triphosphate. And then it spreads out through the rest of the cell, spreads out through wherever it's needed, and then the ATP can release one of its phosphates and give off the energy necessary for wherever it is to actually use. So, for example, the ATP has potential energy. When it moves back into an ADP, a diphosphate, by releasing one of the phosphates, that causes kinetic energy. As in forming an ATP, in other words, taking one with two phosphates and cramming in a third one, is an endothermic reaction because it requires energy, whereas releasing it and releasing energy is an exothermic reaction because it releases energy. Okay. During photosynthesis, as an example, plants use the sunlight, which is an exothermic reaction. Of course, when the sun itself shines, that's an exothermic reaction because it's giving off energy, common sense. And it produces high-energy glucose molecules from low-energy reactants. The photosynthesis reaction is actually as follows over here. I'll just show you quickly. I don't want to get into this too much, but basically you get carbon dioxide and water. The plant needs that. It gets carbon dioxide from the air, it gets water from the ground and its roots from rain, plus energy from the sun, and it turns it into sugar and releases oxygen out into the atmosphere. Oxygen is a waste product, a byproduct of photosynthesis. So what it's doing is it's taking energy from the sun, and it's taken the carbon, actually you need 6 of these and 6 of these to create a glucose. And you're combining the carbon and oxygen from the carbon dioxide with the hydrogen from the water, making CHO, carbon, hydrogen, oxygen, which in these amounts, in the 6:12:6 ratio, is sugar. So this represents organic matter in plants. In other words, this is the thing that has potential energy. So what you're doing is you're taking things without potential energy, and you're using energy to make it into something with potential energy. By the way, so this photosynthetic reaction, this photosynthesis, can you tell me whether it's an exothermic or an endothermic? Now, obviously, you're not going to hear me, but if you want to pause it and think about it and let yourself know what you think it is, I'll tell you what it is in a moment. And of course it is an endothermic reaction. It's not giving off energy, it is absorbing energy. Carbon dioxide and water needs energy in order to become glucose. It's taking energy and it's storing it as potential energy in glucose. It's adding energy, and therefore that is an endothermic reaction. Okay, in fact it says over here on the slide, endothermic synthesis of high glucose water, of high energy glucose, which means sugar, and by taking carbon dioxide and water. In chemistry in general, you need a catalyst in order to establish a chemical reaction. A catalyst is something that increases the chances that a reaction will happen or facilitates a reaction. So a catalyst moves up the rate of reaction without being permanently altered by the reaction. Catalysts can be things like heat or other types of chemicals. It could be a catalyst could be a wide variety of different types of things. So catalysts can speed up reactions by lowering the activation energy. Remember, you need activation energy in order to establish any kind of reaction, but certain catalysts, high as other types of molecules, things like that, can allow something to be created a little bit more, can allow a reaction to be activated more easily. An example, the hydrolysis of sucrose to glucose and fructose. Hydrolysis, remember, means breaking something up by adding water to of a disaccharide, something that has 2 sugar molecules in it, to a monosaccharide like sucrose or fructose, is spontaneous. But if you dissolve it in water and keep the solution at room temperature, it will not happen unless there is at least some sort of catalyst to lower the activation energy. In other words, some things just simply won't happen. There won't be enough energy for them to occur unless you add some sort of a catalyst to make things more to allow things to dissolve or allow things to react more so than they would otherwise. Catalysts can speed up both kinds of reactions. endothermic and exothermic, but again, remember, an endothermic reaction, which requires energy, still requires some energy. No matter how much of a catalyst you add to it, it's still got to have some energy in order to actually form the reaction. The things that are catalysts inside your body, and really the entire point of catalysts as far as this course goes, is the enzymes. An enzyme are protein molecules, and they are in each of our individual cells. They're all over our body. There are millions and millions and maybe billions of them that are floating around our bodies, and they're in each individual cells, and they are probably, well, more than billions. Billions is a dramatic understatement. There are many, many. Anyway, there's a lot. Enzymes are proteins that are in the cell that allow materials to be broken apart, put back together, and various other types of chemical reactions that are necessary in order for the life processes to be done. Enzymes are made of proteins, and they are highly specific for molecules to which they attach. The molecule to which they attach are sometimes called their substrate. Substrate means something that is worked on by an enzyme. The enzymes, in fact, are usually named for their substrate. The enzyme that works on glucose is called glucase. The enzyme that works on DNA is called DNAase. And the enzyme that works on lipids is called lipase. Basically, they just call it with an ASE at the end, based on whatever its substrate is. An enzyme could be used many times for the same kind of reaction over and over again, and it could be regulated by other factors. They have to have the correct site. They have to have the enzyme has to be the same shape. We'll see the diagram in a minute. And they cause the reactions on their substrates, which are the molecules that are reactant, which are the molecules that actually act. The substrates and the active site change shape, and they form what is sometimes called the lock and key model. Now that means nothing in a vacuum, but it should hopefully mean something when you look at a diagram like this. Over here, you've got the enzyme and the substrate, and note that they fit into one another. They call that the lock and key model because it's like a lock fitting into a key. You have the substrate, the purple thing, fits perfectly into the enzyme. The enzyme is specifically designed to have the correct shape to react with this particular substrate. This is an example of a full reaction where the enzyme needs to break apart this substrate. The substrate is the triangle combined with the circle, and eventually you're going to want to break it apart. And the way that works is they have to fit perfectly into the enzyme. The enzyme then performs the reaction by breaking it apart, and there you go. And the same thing is true of the opposite, where you have two substrates coming in here, and they eventually combine to form one product. Now, enzyme activity is, of course, necessary for human life, but they are influenced by certain factors. Poisons and some drugs sometimes inhibit enzymes functioning. In some ways it could be good if the enzymes are overreacting and doing things that are making people uncomfortable or unhealthy, but more often than not, inhibiting enzymes is a bad thing because enzymes cannot do the work that they need to do. Proper pH levels, which is acidity. Acidity is usually measured on a pH scale of 0 to 14, with 0 being most acidic and 14 being most basic, which means the opposite of acidic. Also, temperature is another example of something that impacts the ability of the enzymes to operate. Salt concentration, again, other things necessary, all that impact the ability of enzymes to operate. As far as temperature goes, and the reason why, first of all, acidity, cellular enzymes work best at a pH level of 7.3, which is about neutral. So if you get too acidic inside your body or too basic inside your body, that's not good. Most human enzymes work best at about 98.6 Fahrenheit, 37 Celsius. That's normal body temperature. If you get too hot, you have a fever, or too cold, that's also not good, principally because the enzymes aren't going to be able to work as well. And in essence, when the enzyme's conditions are outside the normal range, they become denatured, or denatured. Sorry if I'm pronouncing that wrong. But anyway, they become a little bit off-kilter in terms of their shape, and they don't work as well. For an example, when an enzyme normally has a maximum activity at 57, but once it's heated to 150 and back down to 57, it still might not work, because if it's heated up too much, it could be distorted. So in other words, just because you bring it back to to the way it was originally doesn't mean you automatically heal the enzyme. The enzyme might have been, think of a crayon. If you heat it up too hot and it melts, and then you cool it back off, it'll refreeze, but it won't refreeze into the same shape as was originally. And remember, enzymes require their shape in order to properly react for their substrate. This is an example of the reaction of different types of enzymes. This is just one example. Let's say you have over here a graph. Let's say this is pH level, 0 being most acidic, 14 being the opposite of acidic, which is basic. And the orange is probably most enzymes in the human body that they react terribly at 0. Then when you go up to 7, they react very well at a little over 7. And then as you get further and further away from 7, they get worse and worse. But then there are other types of enzymes that might work, or other types of reactants that might work better in much lower acidic, much more acidic, or much less acidic environments. And inhibition means you're stopping the enzymes for whatever reason. You're stopping the enzymes from functioning. It could be based on competitive inhibition, which means something else is reacting with those substrates just as well. In other words, let's say if you want some sort of a medicine that stops enzymes from reacting in a certain way, one way to do it is to introduce something into the cell that is going to lock on to those same substrates, but just not do whatever the enzyme would do. Now, if that happens, the substrates just aren't going to. There aren't going to be as many substrates available to react with the enzymes. That's one way to establish to use something to block enzyme activity. And you can also use non-competitive inhibition, which binds to the site of the enzyme. In other words, competitive inhibition is when you bind to the substrate to just use up all the substrates, or you can bind to the enzyme to take the enzymes out of action, because once the enzymes are bound to something that is not the substrate they're supposed to react on, they're not available to react on the substrate itself. Okay, so let's take a look at some multiple choice for the end of this chapter. Which of the following does not have much potential energy? A. A water drop at the top of a waterfall. That sounds like it has a lot of potential energy, right? Because as it's going down the waterfall, it's going to release a tremendous amount of energy as it's going all the way down, so that certainly has a lot of potential energy. A glucose molecule does have a lot of potential energy, because remember, it is energy stored in it that the body can use in order to establish energy for the body. See, a bowling ball that has stopped rolling down the lane. Well, it's on flat ground, and it's not rolling, which means it really doesn't have any potential energy, so that seems right. A diver on a springboard certainly does have potential energy, because he or she can jump off the diving board and go to the ground and very fast. And a AA battery certainly has a lot of stored energy. It can be used for energy. So the only one that does not have potential energy would be choice C. And next question, which of the following is the best description of a chemical reaction? Two reactions that occur simultaneously. Well, again, there could be coupled reactions, but they don't necessarily have to be. That's not much of a description. A reaction that occurs right after another reaction. Again, not necessarily. It could be a sole reaction. Two reactions that occur in the same organelle, same problem. Reactions that occur during sexual reproduction, nothing to do with anything. A process that forms or breaks the chemical bonds that hold atoms together. Yep, that sounds like a reaction, and so therefore that seems like it's the only one that answers the question, and therefore the answer is choice E. Lesson 5 deals with harvesting energy. It means actually using energy in the cell. Now, a lot of this chapter is going to be very technical and, quite frankly, fairly challenging in terms of the large number of different facts and procedures that are listed in the textbook and in this PowerPoint slide itself. What I'm going to try to do here is I'm going to try to just give you the basics of what you need to know, rather than all the information in here. We're not going to expect you to memorize every single term over here, but we do expect you to know the basics of the process. Cellular energy, which means energy on the level of the cell, within the cell, the things that the cell needs to do in order to survive, is necessary. If we don't have cellular energy, the cells can't do anything. We couldn't last more than a few minutes. We could last maybe a couple of minutes without cellular energy, and after that the processes by which the cells operate wouldn't function, and once they wouldn't function, we wouldn't do things like beating our heart and creating nerve impulses to go around the body and things like that. And, well, frankly, we would be, dead. So we don't want to do that. So how do we avoid that? By producing cellular energy. There are multiple cellular reactions that transfer energy from energy storage molecules to energy carrier molecules. Materials that use the energy generated by our food to store energy, and then those molecules transferred to where they are needed to go. The example we discussed before, and we're going to be discussing again, is the ATP molecule. Now this is a quick diagram of the ATP-ADP cycle. Notice both molecules are composed of an adenine group. Adenine group is this thingamajig over here with nitrogen and hydrogen and whatnot. Notice both of these have one of these. That's really what's called ATP and ADP for adenosine, which means an adenine group. And the only difference is how many phosphate groups each one has. Phosphate group consists of a P, which is a phosphorus, with oxygen and hydrogen around it. So you can see the ADP, the adenosine diphosphate, which has an adenine group and two phosphate groups. And then on top you see the ATP, the adenosine triphosphate, which has an adenine group over here and three phosphate groups over here. And the cycle works fairly simply. You have, let's say you start with an ADP group, a regular adenosine diphosphate, and then this procedure, which we're going to learn about in this chapter, glycolysis, causes it to become an ATP. It adds a phosphate group. It uses energy that is received from food that we eat to add an extra phosphate group, to take this extra phosphate group and kind of add it to the ADP to make it into an ATP. Then the ATP is sent out to various parts of the cell wherever it is needed, and wherever it actually is needed to draw off energy to produce energy. And at that point, wherever it is needed, it's the process by which it releases one of the phosphates causes the needed energy to release, and whatever part of the cell needs the energy gets the energy in that way. But then it loses the phosphate group, and then it becomes back an ADP. And then it gets sent back to the mitochondria. Remember, the mitochondria is where this happens. Where this process, by the way, the process by which the ADP is converted into an ATP by adding the phosphate group is called respiration. And that happens in the mitochondria. And so it's a really simple cycle. You release the phosphate group to release energy where needed, and you add the ATP by adding energy in order to store it. The ATP, by the way, of course, has the potential energy, and the kinetic energy, the actual energy, is being released at this point in the diagram, where it's releasing energy and converting into an ADP, which has, of course, much less potential energy. And this process, the glycolysis reaction, this part over here, where you're making an ATP, you're taking an ADP and adding on a phosphate group, how is that done? How does the cell actually convert the ADP? Where does the cell get the energy to convert the ADP, into the ATP? Where does the cell get the energy to do this little addition over here? Well, the answer is it gets it from breaking down nutrients. Nutrients could be fats, and they could be carbohydrates, and could be proteins. Metabolic poison, which is obviously a bad thing, is the sort of thing, one of the things that poison can do is it can interfere with glycolysis when the poison has a structure that's similar to glucose but can't be metabolized. You eat something that the body thinks is glucose and tries to metabolize, tries to break down, and it can't do it. In the meantime, the body's wasting all this energy, the body's wasting all this time, all this glycolysis over here on molecules that are not able to release energy, and it doesn't have enough time to do the glycolysis that is necessary for the body to survive. That's one way in which poisons, of course, can be fatal. Now, glycolysis and the formation of ATP can be done with oxygen, or it can, in fact, be done without oxygen. The difference is, and we're going to look at this in a little more detail, is that aerobic respiration means respiration using oxygen, and anaerobic respiration means without oxygen. The practical distinction is that aerobic respiration can produce 18 times the amount of energy, the amount of ATP, than can anaerobic respiration. So obviously we want to do as much aerobic respiration as possible. Okay, but we'll get to that in a little while. Let's just look at glycolysis. Again, we're not going to focus too much on the minutiae over here. Glycolysis breaks down the 6-carbon glucose. Remember, all sugar, or the sugar that we're dealing with over here, has 6 carbons to release chemical energy. Glycolysis breaks down glucose for energy. Okay. Glycolysis has 2 basic stages. The first stage is the stage that converts the sugar, or uses the energy from the sugar, to convert the ATPs to the format that they are necessary in order to complete the respiration. And then the second stage is actually able to use that energy to convert new ATPs. Long story short, without going too much into detail, the first stage costs 2 ATPs. In other words, it uses up 2 ATPs, which are kind of like units of energy. When I say ATPs, I mean the formation of adenosine triphosphates. So, and then it forms, or the end result is that you have 4 of them. Essentially, when you undergo this stage of respiration, you get 2 ATPs. Congratulations. Now, for each molecule of glucose that is converted into the substance called pyruvate, you've got 2 ATPs. And that could be the end of the reaction. In fact, you can see this diagram over here with glycolysis. You need 2 ATPs, and you end up getting 2 additional, or 2 net ATPs out of it. You have the glucose, and you end up getting 2 ATPs. You also end up getting these 2 extra things called pyruvic acids. 2 ATPs and 2 pyruvic acids. If you don't have any oxygen available, well, that's pretty much the end of the glycolysis. What will happen is you'll have 2 ATPs, and the pyruvic acid that's left over will then either turn into lactic acid or ethanol. Either way, the body can't really use it. The body will eventually have to get rid of it. There are some things that we use lactic acid and yeast for, things like having bread. Actually, well, in yeast. Yeast is a kind of bacteria, or a kind of fungus, so the yeast doesn't actually form within us, but in yeast it turns into ethanol. Again, that's what we used yeast for. Is a little bit beyond the scope of the course, but the point is that at this point, the first stage over here with glycolysis, you're going to form 2 ATPs, and the rest of the pyruvic acid is going to get wasted and turn into lactic acid, turn into ethanol, turn into something that's pretty much not usable. Now here's the bad news, and that's not enough. The bad news that's not enough. If we as human beings were only able to produce 2 ATPs for each for each molecule of glucose that we processed, it wouldn't be enough to survive. Those 2 ATPs are just simply not enough energy to give us what we need to survive. What we do then, but fortunately what we are able to do then, is we're able to go ahead and take the byproducts of the first part of the respiration and use oxygen that we have from breathing in from the outside to convert it and to produce a lot more ATPs, to produce a lot more energy over here. Cellular respiration, which means respiration with oxygen, what we discussed before, the aerobic respiration, breaks down the pyruvate molecules. Remember, before, in the anaerobic phase, the first phase of respiration, we got 2 ATPs, congratulations, and we got 2 pyruvic acids over here. If we didn't use the pyruvic acids, well, then the pyruvic acids went to waste. But on the other hand, if we're able to have some oxygen and to use that next stage, we can then use those pyruvic or pyruvate molecules to produce 32 additional ATPs, obviously a lot more. And this is done in the mitochondria. If we're able to do that, then we're able to produce enough respiration for us to survive. I mean, just to give you an example, if, for example, a person doesn't have any oxygen, a person can't breathe, Well, they can still breathe for. They can still live for a few minutes. You can live for maybe 5, 6, 7 minutes, give it maybe not even that long, without breathing. The answer is because your body can survive on anaerobic respiration for a few minutes, but not much longer than that. If you can't breathe, what's happening? You're not getting enough oxygen into the bloodstream. If you don't get enough oxygen into the bloodstream, well, then the cells don't get enough oxygen to do aerobic respiration, and all they're doing is anaerobic respiration. Some organisms, like yeasts, certain types of fungus, bacteria, things like that, are able to survive only on anaerobic respiration. We're not one of them. We and most other animals and need aerobic respiration in order to survive. So to sum up, what are the ingredients for cellular respiration? What do we need for respiration within our cells? First of all, we can use a form of energy. We can use glucose, which we eat through starches or sugars or things like that. We can also get it from fats, which provide a lot of energy. Remember, as we discussed before, whereas proteins and glucose only give you about 4 calories per gram, fats give you about 9 calories per gram, more than double. And other molecules can be converted into energy by entering cellular respiration at the appropriate stage, like fats and proteins. Even though we saw before the aerobic stage of respiration usually starts only with a glucose, it can also be done, the body can also use respiration from fats and proteins. Some weight loss plans include eating low carbohydrate diet, and the logic behind that is that if you don't have any glucose, or you have very little glucose in the body will be forced to use the other things, will be forced to use the fats and forced to use the proteins in a way that's less efficient, in a way that if you want to lose weight, you want your body to be less efficient. You want your body to have to burn more food in order to lose weight, and so the logic behind, let's say, the Atkins diet or low carbohydrate is that the body shouldn't be able to do normal respiration. It would have to do respiration that's less efficient, that starts at a little bit of a later stage in the process. You can still survive, obviously, but the body will have to consume more energy for the same amount of respiration. Now, chemiosmosis is that last stage of cellular respiration, that last stage of aerobic respiration. This is the process by which energy is used to generate a concentration gradient of positive hydrogen ions, and some of that energy is captured in ATPs, in the adenosine triphosphates, as H-positive ions, hydrogen ions, flow down its gradient. What does that mean in English? Essentially, this is really the key part of respiration, because remember, the original part of respiration is only getting you 2 ATPs, not nearly enough. The thing that gets you all these ATPs, the thing that's so strong that's enough to power the human or the animal body, just with 1 molecule of glucose getting you these 36 ATPs, 2 of the original, it says over here, as we saw before, 32 to 34 ATPs, which means that 1 molecule of glucose can get you 36 ATPs altogether. 2 in the original stage and then up to 34 in the second stage. 36 ATPs obviously is a lot of energy. And the way it does that, long story short, is it generates these ions, these hydrogen positive ions, in a high enough concentration. You can see over here, this is a kind of a very simple representation, where you have these hydrogen ions in high concentration over here, and that naturally causes them to move from one place to another within the structure. And this movement comes with a lot of force and a lot of energy, and that's what ultimately causes all of the energy that allows these 36 ATPs to be formed. Remember, all an ATP formation is doing is taking a phosphate group and using enough energy to weld it into an ADP to make it into an ATP. And then once it's done, the ATP, these 36 or 34 ATPs that are created, diffuse out of the mitochondria back into the cytoplasm, which is the rest of the cell, and then it can go to the rest of the cell or to other cells that need the energy. One of the things that cyanide does, and cyanide could be a deadly poison, could kill people. The way it kills people is cyanide inhibits the enzyme in the electron transport pathway. If the transport pathway is blocked, if this pathway, let me bring that back up on the slide over here. If this pathway over here is blocking these hydrogen ions from moving from here to here, well then not enough energy is being established. And if not enough energy is being established, then the person cannot produce enough energy to keep on with the life processes. And finally, we have the issue of fermentation. Fermentation is the process of what happens to the pyruvic acid if there isn't any oxygen. There isn't any oxygen to continue the process of aerobic respiration. In other words, over here, you do the glycolysis to produce the 2 ATPs and the 2 pyruvic acid. If you have oxygen, you go to aerobic respiration. Otherwise, you do this stage, and this stage over here is known as fermentation. So glycolysis can be done in anaerobic conditions, in other words, without oxygen. And then the second stage is the breakdown of the pyruvic acids into lactic acid in something known as fermentation. The pyruvate in the cytosol, in other words, that has not yet been converted, has not yet been acted on by oxygen. It can't be. After a while, it just sits there and does nothing because there's no oxygen there for it. And it can either produce lactic acid, which is usually done in human beings, mammals, and certain bacteria, as you can see in this diagram over here. And in yeasts and certain other organisms, instead you produce ethanol, which is really a type of alcohol. So, and then in humans, you have in mammals and bacteria, you have the lactic acid that's being formed. That's the left side of this document. Now, when the lactic acid is being formed, that cannot be used. It just kind of builds up and is really a waste product in the human being. The muscles become sufficiently low on oxygen to perform glycolysis. Basically what happens is when a person is exercising, a person's running around, a person's biking, whatever it is, the body says, we need more ATPs, we need more ATPs, we need more ATPs because we know we need more energy, and the body starts respirating as many glucose molecules and other molecules, fat molecules, protein molecules, whatever it is, as possible. Then, the thing is, if you breathe in, no matter how much you breathe in, you might not have enough oxygen to get 36 ATPs for each molecule of glucose that you're respirating. And so because of that, kind of the pyruvic acids from all of this respiration builds up. And this causes fatigue, and it also could cause some discomfort as well. So during vigorous activities, muscles become sufficiently low on oxygen, and they do more glycolysis, and more really respiration to produce more ATP molecules, which produces a brief burst of speed. However, if there's not enough oxygen in the body, which is, by the way, also interestingly enough, why we breathe fast. When you run and when you get out of breath, when you breathe, you're breathing very quickly. And the reason why you're doing that is because the body's saying, hey, we need more energy. In order to get more energy, we need more respiration. In order to do more respiration, we need more oxygen. So the brain sends a message to the lungs, to actually really the diaphragm, and says, get us more oxygen, get us more oxygen. That's why you breathe very quickly in order to try to, you're kind of panting because the body's saying, get us as much oxygen as possible. So when a person bicycles up a hill, you feel a burn in your legs or fatigue in your legs. The reason is because your legs aren't doing enough cellular respiration. Instead, you're doing anaerobic respiration, and the lactic acid is building up in your legs, causing a little bit of discomfort. The same thing is true with bacteria. Bacteria also does anaerobic respiration, and bacteria in the mouth feed off the sugars that we eat and produce lactic acid, which cause cavities and also bad breath. The bacteria in our mouth break down the sugar and do anaerobic respiration, where bacteria and microorganisms like that don't do aerobic respiration, they only do anaerobic respiration. The other type of fermentation is done by yeasts and certain other materials and certain other types of microorganisms, and this is the right side of the diagram, the ethanol or alcohol fermentation. So in this type of scenario, when it's done by yeasts and certain types of microorganisms, the pyruvic acids are converted into alcohol and carbon dioxide, into ethanol and carbon dioxide. And that, of course, is why the carbon dioxide actually caused bubbles of carbon dioxide, which is why bread rises. Bread rises because you have the wheat molecules, or you have the bread molecules, and you have little pockets, tiny little pockets of carbon dioxide that blow them up and make them bigger and softer because they're more spread out, and that's what causes bread to rise. And it also is what causes alcohol to form. Grapes and things like that are aged in an area where you have these yeasts, you have these microorganisms that do aerobic respiration on the sugars in the wine or the sugars in the grape juice, and the pyruvic acid that they make ferments into ethanol, ferments into alcohol, and that's what eventually causes wine to form. So when yeast ferments, it produces carbon dioxide, which causes bread dough to rise, and a single yeast cell undergoes alcohol fermentation and uses 50 molecules of glucose to only produce 100 molecules of ATP. Basically, that's a review of what we discussed before, that fermentation, or non-aerobic respiration, produces only 2 ATPs per molecule of glucose, whereas aerobic respiration produces up to 36. And now let us look at some of the a couple multiple choice questions for this chapter. Okay, so let's look at the questions. In the absence of oxygen, suppose a single yeast cell undergoes fermentation and uses 250 molecules of glucose. How many molecules of ATP will be generated? Well, as we know, this is anaerobic respiration because it's a yeast cell which does only anaerobic respiration, not aerobic, and so it's only going to produce 2 molecules of ATP per molecule of glucose, so that's 250 times 2, which is, of course, 500. Easy enough. 2. Cyanide poisoning occurs because cyanide inhibits an enzyme in the electron transport pathway. Which of the following is the reason why the cyanide poison becomes deadly? Well, we discussed this earlier. A. Cells switch to anaerobic fermentation. That seems to be correct initially, because it can only do anaerobic. It can't actually now use the oxygen to do aerobic respiration and produce the extra ATPs. So let's see the others. Oxygen is reduced to water. I don't know what that means. Glycolysis stops. That's not totally true, because glycolysis is the first stage. Glycolysis is this stage where the pyruvic acid is formed, and that's not really what cyanide inhibits. So that's not correct. And ATP is no longer chemiosmosis. That actually seems to be correct. That's actually a more correct answer. Yeah, that's the chemiosmosis. Well, that's what cyanide does. It tries to do it tries to do the aerobic respiration, but doesn't quite do it. So A is actually a superficially attractive choice, but I think D is the better choice. So we're going to choose D over here, and I will see you at the next section. The next lesson, lesson 6, is about reproduction and genetics. The first thing is cell division. Cells have to reproduce one after another. If you have one skin cell, well, then it reproduces into many skin cells. That way, when skin cells die, the new ones form. They say that you get a brand new body once every 6 weeks or so, because all the cells in your body at one point or another die and then are recovered, but replaced by other cells. And the process by which this is done, the process by which normal cellular reproduction happens, is called mitosis. M-I-T-O-S-I-S. And mitosis works very simply. You've got an original cell over here. You can see these things in here are the chromosomes. A human being actually has 46 chromosomes. This diagram only has 2, but that's okay. You can imagine it. And what happens in the process of mitosis is the cell it doubles itself. Then the cell itself splits apart, and you end up with 2 daughter cells that are the same as the parent cell. 2 diploid cells, which means double, that have the same number of chromosomes as the original. In terms of organizational reproduction, in other words, the entire organism reproducing, you have sexual and asexual reproduction. Sexual reproduction simply means you have 2 cells, one from 2 different organisms, one from a male cell and one from a female egg cell, and they combine to form an offspring. Asexual reproduction is simply where you have one offspring that gives rise to another one. Let's look at them one at a time. Mitosis, which we just looked at, this is the regular cell division that happens in your body, and the way that small cells like bacteria and things like that split apart into two daughter cells. Diploid cells, which means again cells that have the same number of chromosomes as the parent cell, have a pair of homologous chromosomes, which means chromosomes that go together with each other. I mentioned before that human beings have 46 chromosomes, and that's because we have 23 pairs of chromosomes. 23 they're not twins, they're different from each other, but 23 pairs of chromosomes. Meiosis is how we produce our reproductive cells. Our reproductive cells only have half the number of chromosomes as the parent cell. So, for example, in an organism like a human being or a mammal or even most trees, you have a male reproductive cell known as a sperm cell and a female reproductive cell known as an egg cell, and each of them have half the number of chromosomes. So, for example, in a human being, a sperm cell and an egg cell will each have 23 chromosomes, whereas a full organism has, of course, 46 chromosomes. Here's a little diagram like the one that we just saw. We've got mitosis on the left side, which is a parent cell. It replicates and it comes out to two equal daughter cells. Whereas over here you have meiosis, where a reproductive cell is being formed, and notice the daughter cells only have half the amount of chromosomes as the parent cell, and eventually you have a process of fertilization where the two cells join, and this over here is what is called a zygote, which means the first cell in a brand new child. Here's a little bit of a bigger diagram over here of meiosis, where you have a parent cell, DNA replicates into two daughter cells, and each process produces four reproductive cells that can then be used to reproduce eventually. Now, of course, there are some problems that exist that when things go wrong. One very famous example, is the example of Down syndrome. Down syndrome is, as we discussed before, there are 23 pairs of chromosomes, and Down syndrome occurs when through some sort of a problem with the reproductive process in meiosis, an extra copy of chromosome 21 is there, and it affects about 1 in every 700 people, and people who are affected by Down syndrome very often have mental retardation problems and shortened lifespans. This happens when one pair of chromosomes fails to separate during meiosis. Remember, during the meiosis process at the beginning over here, really in this second stage over here, the chromosomes split into half the number as the parent already had. Now, if one of them fails to split, so instead of the daughter cells, let's say, having 23 chromosomes each, they each have 24 chromosomes, then you're going to have too many chromosomes in the child. Instead of having 46 chromosomes, the child's going to have 47, and the problem with the 21st chromosome not splitting is something that specifically leads to Down syndrome. Here's really the example. You have normal meiosis, is where they split apart to have half the number of chromosomes, as you can see over here. If I know it's very small, I'm sorry for that. But what happens in the right side, in the Down syndrome case, is 24 chromosomes split normally at the beginning over here, but look what happens to the second stage. Instead of being only on this side would be the normal area, one black and one orange on both sides. Over here you've got two blacks and one orange, and this cell eventually goes to fertilize and become a child, and that child could have Down syndrome. Now we're going to move to genetics. Genetics is the study of how characteristics are passed from one generation to another. You probably have noticed that parents who are tall tend to have children who are tall. Parents who are smart tend to have children who are smart. Parents who have darker skin tend to have children who are darker skin. It's because of the idea that parents pass their DNA. The parent's DNA goes to the to the offspring. About half of the DNA comes from the father and half of the DNA comes from the mother. And the reason is because diploid organisms, which means organisms that have two pairs or pairs of chromosomes, have pairs of, well, pairs of chromosomes. Homologous means they form pairs. So we have 23 pairs of chromosomes. Each of our 23 pairs, one of the chromosomes came from our father, and one of the other chromosome came from our mother. So they have two copies of the same gene. If both have, now an allele means something that expresses a particular trait. Let's take, eye color. Eye color, there are three basic eye colors. There's blue, there's green, and there's brown. Hazel is a combination of green and brown, but those are the three basic, most common eye colors. Now, an allele would be, let's say, for example, a gene for a green eye. So if you have two genes for green eyes, your mother gave you a green eye gene and your father gave you a green eye gene, then the organism is what is called homozygous. Homo meaning the same. It's the same. They both have both alleles, both genes are the same. Both are for green eyes. If the homologs have different alleles, the organism is heterozygous, which means it's mixed. You might get one green eye gene from your mother and a blue eye gene from your father. Now there is a distinction. You have one green and one blue. Now we'll discuss in a minute which one you actually get, but it's heterozygous because you don't have the same one from both parents. A genotype means what the gene actually is. If you have a green eye gene and a blue eye gene, then your genotype is one green and one blue. Which one is actually expressed is called the phenotype. For example, let's say a cell in a person's eye has the genotype for brown here, blue eyes, not have hemophilia, and a lot more information. Excuse me. In this example, this is really based on this similar idea that every cell in your body has the DNA to express every gene in the entire body, but only some of them actually are expressed. The gene in your eye has DNA that tells your feet how to behave too, but only expresses it based on only is relevant to your eye. I'm sorry, I skipped a couple of slides over here. We'll just go over this. I mentioned this already, but let's mention it again. Genes, a gene is a segment of DNA that are the units of inheritance. DNA is made from proteins and produces proteins, cells, and really the entire organism. Every cell in your body has the DNA really to express everything in the entire body. Locus is the gene's location on the chromosome. At a particular point on the chromosome, you have the gene for your eye color. At a different point in the chromosome, you have a gene for your hair color. At a different point, you have a gene that illustrates your height. And an allele could mean different versions of the gene at a given point. A trait or an allele could be dominant or could be recessive. Dominant means it shows up, and recessive means it doesn't show up. Just to give you one example, blue eyes are recessive as to brown eyes, which are dominant. A person has two brown eye genes, they're going to have brown eyes. A person has two blue eye genes, they're going to have blue eyes. But if a person has one brown and one blue, they are going to have brown eyes because brown eyes are dominant and blue eyes are recessive. Same thing is true with green and blue. If a person has a green and a green, they're going to be green eyes. A blue and a blue is going to be blue eyes. But a green and a blue, they're going to have green eyes because green is dominant over blue. Green and brown are actually codominant. That's why if somebody has a green and a brown eye gene, they're going to have hazel eyes, which is kind of a combination of green and brown. This is the way, basically, a pair of chromosomes look. You got two chromosomes that are a homologous pair, and on each point on the gene over here, you could have two alleles that go for the same thing. Like this green area and this green area both establish hair color. They could be the same, in which case you have the homozygous trait, or they could be a heterozygous trait. And again, as we discussed before, the genotype is what you actually have in terms of the gene. The phenotype is how the gene is expressed physically. You can also use what are called Punnett squares to make a determination of what the odds are of a particular child of particular parents having a particular gene. This is based on the idea of what is likely, what are the odds of a particular trait being passed to children. Basically, I mean, I'll give you examples over here, but the easy way to draw the Punnett squares is to draw a box with 4 squares, and for each possibility, you draw them on top and on the side. Well, I'll put that as an example over here. Without an example, it's impossible to figure out what that means. Let's take an example. A father has one dominant and one recessive gene for brown eyes, and the mother has one dominant and one recessive gene for brown eyes. We'll call big B brown and little b blue. What is likely for the child to have? Now, for this Punnett square, you put one parent up here and one parent on the side, and the father has one big B for brown and one little b for blue. That's the father's genotype. And the mother's genotype is also one big B and one little b. So you kind of draw a little bit of a grid over here and see what the combinations establish. This box is a combination between this and this, which is 2 big B's. This box is a combination between this and this, which is 1 big B and 1 little b. This box is a combination of this and this, which is 1 big B, 1 little b. And this box is a combination of this and this, which is 2 little b's. So we see over here there's one homozygous brown, one homozygous blue, and 2 mixtures, 2 heterozygous genes. Now the 2 heterozygous genes are both going to end up with brown eyes because brown is dominant. So in this example, if you have a father that's heterozygous brown and a mother heterozygous brown, the odds are going to be about 75, since the chances are equal that the father will either give a big B or a little b, and the chances are equal that the mother will either give a big B or a little b. The odds are 75%, namely 3 out of 4, that the child will have brown eyes, and 1 out of 4, namely this guy in the corner over here, that the child will have blue eyes. If we had, other examples, let's say, for argument's sake, you're talking about, let's say, again, let's try capital B for brown. Let's take this example where a capital B is brown, a G is green, and a B is blue. Remember, B and G are both dominant because they give you hazel, but either one of them is dominant over blue, which is recessive. So let's say, for example, you've got a father who is brown heterozygous and a mother who is green homozygous, in other words, 2 greens. Now, if you wanted to know what the child would be, then you would draw a little Punnett square over here, and you would take the 4 combinations. In this box, you've got a B and a G. So this is, well, let's do all of them. This one is G and B. This one is also BG, and this one is also GB. So these two would both be brown and green, which are codominant, and you'd have hazel. That's 50%. Half of them are hazel. And this half would be green and blue, which turns out to be green. Also half of the children. Let's take a different example. What if instead of, I'll tell you what I think just my own family, I believe, is that, I mean, I have particular reasons to believe this, but I think my father happens to be heterozygous brown, so a brown and a blue. My mother, I believe, is heterozygous green, which is a green and a blue, which means in my family, what do we got? We got 1 chance for BG, which is hazel. That's what I have. We've got a GB over here, which is green. My sister has that. We've got a BB, capital B, lower b, which is brown and blue, which is brown. That's my brother. And the last one is BB, blue blue, which is possible for a blue-eyed, and that's none of my siblings. I've got two siblings, so we've got this one, we've got this one, we've got this one, but we've got no blue-eyed. But again, these are just odds. It doesn't mean that if there was one more child in the family, the child would definitely be blue eyes. These are just based on the odds. And in case you're curious, the reason why I'm pretty sure that my sister is green and blue is that when she was a kid, she always had blue eyes, and they turned greenish as she became older. I think that what that means usually is that you have a blue eye gene, and it takes the green eye gene a while for its dominance to assert. It takes a few years for its dominance to assert. Family charts and things like that, something known as pedigrees, can actually help you see which types of genes, specifically diseases, very often used for, are carried in a particular family. Now, this chart happens to be, it looks like a royal family, looks like a German and British royal family over here that happen to have a lot of hemophilia in the family. Hemophilia is a disease that the blood doesn't clot properly, and it could be very dangerous if you get a cut, get a deep cut, something like that. You could actually bleed out from it. And this actually, the disease happens to be on the X chromosome. And males, we didn't discuss this before, but a male has a X chromosome and a Y chromosome, whereas a female has two X chromosomes. And the disease is recessive, which means if you have one good X chromosome and one bad X chromosome, you do not have the disease, which means it's a lot easier for men to get the disease than women, because women have two shots at a good X chromosome, and men only have one shot at a good X chromosome. If a woman has one X chromosome with the disease and one without it, the woman is called a carrier, because she has the X chromosome for the disease, but she doesn't actually show the disease. She's not actually sick. Again, just the point is, this is an interesting chart where it shows the clear boxes are men who do not have hemophilia. The black boxes show men who do have hemophilia. The white circles have women who don't have the gene, and the half-shaded circles show the women who are carriers, who have one X chromosome with hemophilia and one without hemophilia. This is one example over here. So scientists can figure out who the carriers for a disease are, and who wasn't a carrier, and who had the disease. Sometimes, there's an organization which many of you are probably familiar with called Darya Sharm, and what they do is they try to see who's a carrier for which disease. And if there are bad diseases like Tay-Sachs, familial dysautonomia, and certain other diseases that they test for, if two parents, if only one of them is a carrier for the disease, it's not a problem, because the child can then at worst only be a carrier. Let me draw that example. Let's say you have, let's say an H for healthy, and a C for a carrier. So if you've got two parents and one of them is healthy totally, and the other is a carrier, neither of them are going to have the disease, because the disease is only recessive. It's not dominant, so you won't see it. Then if you notice, the four possibilities for the children are healthy, healthy, and over here is also healthy, healthy. Over here is healthy and carrier, and over here is also healthy and carrier, but none of the kids are sick. At worst, the kids are carriers. On the other hand, if you have two parents who are both carriers, in other words, you've got an H and a C, together with an H and a C, well, 3 out of the 4 kids are going to be okay. You're going to have an HH, which is not even a carrier. Over here, you're going to have an HC, which is a carrier. Over here, you're going to have an HC, which is a carrier. And over here, on the other hand, you're going to have double Cs, which means you're going to have 2 carrier genes, which means the child is going to be sick. Which, if there's a disease like this, like Tay-Sachs, which is a recessive disease, but you have 2 carriers, there's actually a 1 in 4 chance that each child will have the disease. And that's what an organization like Darya Sharm does. They'll tell the parents that this shidduch is not really a match, because the children could end up having these very difficult recessive diseases. This example over here and previously, this example is different than the one I just gave. The one I just gave is where the disease is on just a random gene out there, whereas over here you're talking about a disease that is on the X chromosome. You have the over here, Louis the 14th was not a carrier of a disease. Where's Louis the 14th? I can't even find him. Somewhere over here. Here we go. Louis the 14th, right over here. Then we had Alice, Prince of Hesse or Hesse, was a carrier of the disease, right over here. I guess she married Louis the 14th. And their children, you can see, the children either were carriers, totally healthy, or poor Frederick over here actually had the disease, because he must have gotten the X chromosome. The male cannot get an X chromosome from the mother, from the father, can only get a Y chromosome from the father. Essentially, let me take a few moments to discuss that as well. The way gender determination works is that men have an X chromosome and a Y chromosome, and a woman has two X chromosomes. So when there's fertilization, the male always has one X and one Y, whereas the female has two X's. And you can see the odds if you draw a little Punnett square over here. You've got X and X over here. You've got XY over here, XX over here, and XY over here. So, of course, half of them are men and half of them are women. That's why, of course, the odds of a baby being a boy or a girl are roughly equivalent to each other. Now, if there is a disease on the X chromosome, a boy can only get an X chromosome from the mother. Because if it's a boy, it means they're getting a Y chromosome from the father by definition, which means it must get its X chromosome from the mother. So if the mother is a carrier, like Alice over here, she's got two X chromosomes, one good one and one with hemophilia on it. So Frederick got the Y gene from Louis the 14th, but he got the X gene from Alice, which means he had a 50-50 chance of being sick, which means he was okay, but his brother Ernest, he was not okay. He was a hemophiliac, but his brother Ernest was okay. Okay, self-fertilization, there are some plants that actually have the male and female, even some animals, believe it or not, like sponges and things like that. Well, not sponges, but certain other very, very basic types of animals that have both the male and female parts at the same time. And trees very often do that, and different types of plants as well. And an organism, sperm, fertilizes its own egg and does sexual reproduction, even though it's only one organism. The person that originally did all of these experiments to determine these Punnett squares that we discussed was an Austrian monk named Gregor Mendel. You might remember him from your high school biology class. And he did all these tests. We don't have to go through them in exhaustive detail, but he did all these tests to determine the effect of recessive and dominant genes. Okay, so here are some examples. We did some examples before. I don't think we need to do any more of these examples. Cell reproduction could also go wrong. It's called when cell reproduction gets out of hand and reproduces beyond control and just goes crazy and grows too big and interferes with other cells. That is what is known, unfortunately, as cancer. Mutations, which means some sort of a problem in the mitosis process, in the process of the cells reproducing. Mutations happen all the time. One individual mutation is not a problem. Usually if there's a mutation, some sort of a problem, and it causes a problem with the cell, then the body just recognizes it, crushes it, destroys it, and flushes it out of the body, and that's that. You might have 100 mutations a minute. It's not a big deal because the cell can recognize them, crush the cell. It's only one of many, many billions and trillions and who knows what number of cells you have in your body, so one cell is no big deal. But if it causes uncontrolled cell divisions, you have a lot of cancerous cells, or the cancerous cells start to get out of hand, obviously that could cause a big problem. one of the ways they try to treat cancer is to focus in either with chemotherapy or with radiation therapy to try to kill the cancerous cells. Now, unfortunately, the problem with both of those approaches is that they may kill the cancer cells, but first of all, they might not be able to kill every single one of them, and so it might come back later on. And second of all, those sorts of therapies also attack and kill the healthy cells that are nearby, which causes all sorts of problems to the patient. And let's take a look at some multiple choices quickly over here. Which of the following is represented by frogs? Frogs are advanced life forms, or at least they're animals. Certainly they're not bacteria or fungi or anything like that. And they represent the diploid life cycle, which is mitosis, in the idea that there are two pairs of chromosomes, or pairs of chromosomes, for every chromosome that they have. And so therefore the correct answer is B. Which of the following plays a role in cancer development? Not controlled cell division, which is okay, but mutations is certainly the correct answer. CDKs do not stimulate cell division. Wearing sunscreen is actually good for you. That's certainly not going to cause cancer. The problem that does cause cancer could be mutations, so that's B as well. Lesson 7 is about DNA and heredity. DNA is the substance that we all have in each of our cells within the nucleus, as we discussed earlier, and that is really the blueprint for the entire cell. Gives the ability of all the other cells in the body to carry out their functions. Heart cells to pump blood, eye cells to be able to see, etc., etc. And DNA stands for deoxyribonucleic acid. It's a really complicated word, but if you break it down, it's not so complicated. And really, it's a series of what are called nucleotides. A nucleotide is simply a combination of a base, a phosphate, and a sugar. Most sugars, like the glucose we learned about earlier in the course, have 6 carbons, whereas the sugar in DNA has 5 carbons. So, and a nucleotide, which is really the building block for DNA, has a 5-carbon sugar, a phosphate, and a base, which is a molecule that has nitrogen in it, hence its name, the nitrogen base or nitrogenous base. Here's an example of a nucleotide. Adenosine, we actually saw adenosine before when we were talking about ATPs and ADPs, but it's also an example of a base that could apply in a DNA molecule. You've got a sugar over here. Notice the 5 carbons, 1, 2, 3, 4, 5. And you've got a phosphate group over here, which has a phosphorus in the middle, surrounded by a bunch of oxygens and hydrogens. So that's what DNA is. DNA is a nucleic acid, which means a bunch of nucleotides. Notice that word there. And the reason why it's called deoxyribo is because it is a ribose sugar that's missing an oxygen. We'll see a little bit later on there's something similar called RNA, which is ribonucleic acid, which is not missing an oxygen. Anyway, so it's called deoxyribonucleic acid, and a nucleotide, as we saw, has a phosphate group, a sugar called deoxyribose because it has 1 less oxygen than a comparable sugar called a ribose, and it also has a nitrogenous base. And there are 4 different possibilities: A, G, T, or C. Try to remember the letters. You don't necessarily remember the names. The names are adenine, guanine, thymine, and cytosine. And interestingly enough, it's really just in the order of those bases that makes a determination in terms of what's going to happen to the cell and with the cell. The DNA is shaped in sometimes what is known as a double helix. We're going to see a little bit on the next slide. A double helix is kind of like a spiral staircase with stairs being the bases. Here's some examples of some representations of the double helix. These are just different types of diagrams, different examples. The backbone over here, this line, and in this diagram it would be this line and this line, consist of the phosphate group and the sugars, and these little bars going out to meet each other are the bases. In other words, there's one on this side and one on this side, and this base is moving to meet with that base. That's why it's called a double helix, because there are 2 strands of DNA that are kind of wound around, and they're facing each other, and each nucleotide meets its opposite nucleotide, the one on the other helix, with a base that extends out to the other one. Okay, so let's take a look at the slide. Over here, the DNA ladder is like a spiral staircase with two strands wound around each other, and the bases are held together with hydrogen bonds. They can be broken by some sort of a drug or medication, but again, I don't even want to really go into that right now. But the point is, one important thing is that, notice, as we discussed before, this is the backbone of it, which contains the phosphates and the sugars. And these things over here are the bases. Now, the one thing that is important is that A only bonds with T, and C only bonds with G. So if you can remember those four letters, A, T, C, and G, which is adenine, thymine, cytosine, and guanine, the C only goes with the G, and the T only goes with the A. So for example, in this diagram over here, the green thing being adenine, the yellow thing being cytosine, purple guanine, and I guess maroon thymine. So you can see here is the A and the T, this is the C and the G, AT, GC, GC, AT, etc. You can see the A's always go opposite the T's, and the C's always go opposite the G's. That's the way the double helix is structured. Here's another representation of it. Probably a better diagram over here. On the top just represents the fact that you have the A's opposite all the T's and the C's opposite all the G's. And on the bottom you have the same thing but in the form of a double helix, which is the way the DNA really operates. Kind of two strands winding around each other. Helix is really just another word for a spiral. Now, the thing that DNA is important for is that the DNA is the thing that codes your genes. We're going to see a little bit later on. Each three bases of DNA are able to encode one amino acid. And how that combination, we've got millions and millions and millions of bases of DNA in each of our cell, and that combination, the one that codes the appropriate amino acids, makes the difference between somebody who has brown eyes and somebody who has blue eyes, or between a human being and a fish and a goldfish, is really just the base order that's in our DNA. Now, we happen to have different amounts of DNA also than goldfish do, but because the DNA is what programs the amino acids to do what they do. So, genes are made of DNA, and DNA encodes genetic information. The way the DNA actually works is that it produces two double helixes, or double helices, with the same genetic information on both, and that's how the cell replicates. Remember when we discussed mitosis, we discussed how the chromosomes replicate. The chromosomes split apart into two identical chromosomes, and they each eventually go and form part of the child, part of the offspring. RNA is the partner of DNA. The DNA essentially just stays in the nucleus, doesn't go anywhere. It's kind of like the blueprints that you're not allowed to take out of the library. You can go to the library and look at the blueprints, but you can't take it out of the library. If you want to get a copy of the blueprints, you can photocopy the blueprints in the library and then take it out. Well, that would be the RNA, because the RNA, in essence, photocopies the DNA and moves it around to different parts of the cell, to the ribosomes where the amino acids are being made, and to whatever other parts are to the mitochondria, whatever other parts are necessary to be programmed by the base sequence that apply in the DNA. So the DNA itself cannot actually synthesize the proteins like in the ribosomes. Instead, it uses the RNA to get its message out. RNA, as you can imagine, is very similar to DNA. It is also made of nucleotides. It is also made of sugar, phosphate, and base groups. There are a couple of subtle distinctions between DNA and RNA. You might want to remember these. These could be important. It has a backbone of ribose instead of deoxyribose. Remember, the nucleotide for DNA had a sugar, base, and a phosphate, and the sugar was called deoxyribose, whereas the RNA also has a sugar, phosphate, and a base, but the sugar is ribose. As you might be able to imagine, the only distinction between ribose and deoxyribose is that the deoxyribose has one less oxygen, hence the deoxy. The other distinction is that instead of the T, the RNA has a U, uracil. So instead of the T going with the A, it's the U going with the A. So, and the C and the G are the same. That's just, again, one subtle distinction between DNA and RNA. Now, the way RNA works is essentially what you have. I'm going to skip to the slide before and skip the bottom for the time being. You have two types of RNA. You've got the mRNA, or the messenger RNA, and they basically photocopy or get attracted to the bases of the DNA in the nucleus. Let me find a diagram like that. Here we go. This is a good one. Let me show it to you over here. Okay, this is a classic example of transcription. You've got this stuff, this dark blue material over here, and that's the DNA. Now, you notice how in the DNA you've got lots of bases, A, C, T, G, A, T, C, etc. And they all have the opposite bases over here. T opposite the A, C opposite the G, A, T, C, G, etc., etc. So that was the original DNA double helix. And what happens with transcription is the DNA kind of splits apart. You can see these two kind of came apart. And the bases of the DNA attract RNA bases that are their mirror image. The T's attract A's, the A's attract U's. Remember, there are no T's in RNA. This green strand is an RNA. The C's attract G's, and G's attract C's, etc. So now if you notice, once this happens, you've got an mRNA strand, a messenger RNA strand, and that is the exact mirror image of the DNA, because the bases are the exact opposite. Then the mRNA goes out into the ribosomes, or goes out into wherever they need to be, and they attract another set of RNA called a tRNA, called the transfer RNA. And if you notice, that's really kind of like a double image. The DNA attracts the mRNA that's the opposite of it. Then the mRNA attracts the tRNA that's the opposite of it. And so the tRNA are essentially going to be the same thing as the DNA, which of course, with the exception of the U's being instead of the T's. So the mRNA carries the information from the DNA to the cytoplasm, in other words, to the outside of the cell. And this is kind of a copy of the DNA. And the transfer RNA then gets encoded by the mRNA and essentially is the same coding as the DNA. And then what happens is that each three bases, each pair of three, well, really try a link of three bases, is enough to encode one amino acid. An amino acid is the building block of proteins in general, and a three-base sequence is called a codon. And the reason why it's called a codon is because it encodes one amino acid. And a group of amino acids, when you get a whole bunch of amino acids together, they make a polypeptide, which, as we learned earlier in the course, is really the same thing as a long protein chain. And so then the mRNA is now capable of, the tRNA is now capable of, wherever it is, in the ribosomes or out in the nether reaches of the cell, wherever they happen to be, of encoding these codons. And the codons then determine what kind of amino acids are formed, and what kind of amino acids are formed determine what they're actually going to go out and do. Where problems arise is sometimes referred to as a mutation. A mutation is what happens when a DNA sequence gets messed up a little bit. Base sequences get altered, some of the bases get cut out, some of the bases are doubled, some of the bases are halved, whatever the case may be. Mutations is when there's some sort of a problem. Now, these happen all the time, and they're typically fairly harmless. If it's bad enough for the cell to have a problem, the cell can just be killed off and replaced. Obviously, a lot of mutations, or really bad or really unlucky mutations, could cause serious problems for a person, but mutations in general are fairly routine. So if a base pair is mismatched, then the mismatch can simply be cut out, and it can just be reformed in the correct way. However, sometimes the enzyme replaces the nucleotide instead of parental nucleotide instead of the mismatched one. And so what could happen is, because of a mutation, it turns out there's some sort of a mismatch in the DNA or in the RNA, whatever is encoding the amino acids. And because of that, what that might happen is it just could change the function of the cell. And the cell, it doesn't have the right amino acids and either can't function or functions in a different way. There are different types of mutations. Insertion is when an extra base is inserted. Deleted is when a base is deleted. Inversion is when bases are backwards. And translocation is when they're changed places. And a base deletion near the start of a coding sequence, let's say near the start of the sequence that codes what what's going to happen, what a person's eye is supposed to do, that's obviously the worst kind of mutation. So this is just some examples of different types of mutations. Point mutation is where you have, let's say, the wrong type of base gets inserted over here. Insertion is you have an extra base, this T over here in the middle. Deletion is when this A base got deleted. Point mutation, you can see, these are pretty self-explanatory, and these could cause diseases and could cause serious problems, especially when you're talking about individual like a zygote, a brand new baby cell that doesn't have enough cells in the rest of the body to overcome it. And mutations could cause obviously very dangerous things like genetic diseases and even cancer. And these bases are the format for genetic expression. The appearance of a phenotype, like brown eyes or black hair or whatever it is attributed to a particular gene. In different tissues, different genes are expressed. Even though your DNA in each cell has what is necessary to program the entire body, in your eyes, only the cells that affect eyes are expressed. In your heart, only the DNA that affects the heart is expressed, etc. Because your eyes don't grow hair, because different genes are expressed in different tissues. Your hair-growing genes are not expressed in your eyes, thankfully. Okay, so let's take a look at a couple of multiple choice questions for this chapter. A sequence of bases in the DNA section is AA, TC, GG, AT. What is the corresponding sequence of mRNA? Well, remember, you always have U's instead, it's RNA, so you always have U's instead of T's. So the A would correspond with a U. So just looking at the first three, AAT would have to be UUA. And I can see there's only one choice that starts with the UUA. Let's just make sure the rest are correct. CGG would be GCC, which it is. And then AT would be UA, which it is. So C would be the corresponding example of base sequences to this one. Next one, which of the following has the fewest nucleotides? Okay, well, mRNA strand having 100 nucleotides, okay. mRNA having 75 codons. Now remember, a codon has 3 bases, and each nucleotide has 1 base. So something that has 75 codons would actually have 225 nucleotides, 225 bases. Protein having 50 amino acids. Now amino acids are programmed by 1 codon, so that would actually have 150 bases, 150 nucleotides, so that's more. So far the least is A. A protein with 10 polypeptides having each 40 amino acids, that's 400 amino acids altogether, that's obviously a lot. And an mRNA having 100 bases. I don't know, it looks like, I'm just going to cut choice E because choice E looks like the same as choice A. So anyway, the correct answer, if we cut choice E, is just plain choice A. Our next chapter is on homeostasis. Now, really, the entire body and a lot of living biology, a lot of biology in general, could be put into the category of homeostasis, because all homeostasis means is establishing consistency or constancy within an organism. Obviously, in order to survive, an organism needs to be able to make sure that things stay pretty constant. If something outside makes the organism hot, the organism has to figure out a way to cool itself down. If something outside makes it cold, the organism has to figure out a way to warm itself up. If something outside makes the level in a person's bloodstream, for example, too acidic, well then the body has to figure out a way to counteract that. Making sure that you counteract whatever external forces try to wreak havoc in your body or in the organism is the concept of homeostasis. Stasis, you may recognize, means consistency, or static is another word, making sure that things stay the same. So homeostasis in general means the idea Excuse me. So homeostasis is the ability of an organism to maintain its internal environment that in a manner that allows optimal cell functioning. In other words, in a manner that allows it to function as it's supposed to. That's generally what homeostasis means. How is homeostasis achieved? The answer is through feedback mechanisms. Feedback mechanisms are things within the cell or within the body that produce reactions that either counteract a negative outside influence or stimulate a positive influence. Positive feedback means something causes an increase in something else. Negative feedback, on the other hand, is when the body tries to counteract something. We'll see examples in a little while, but this is one quick example of negative feedback. When the body temperature rises, therefore it produces sweat, and that causes the body temperature to drop. Negative feedback is, of course, a very important mechanism in homeostasis. So when blood oxygen levels are low, you try to breathe in more oxygen automatically. Like one example we discussed earlier in the course, that when a person's running and needs more oxygen for respiration, there's now a deficit of oxygen in the bloodstream because it's being used up by the cells. And so the body says, okay, get us more oxygen, and you breathe in much harder. It also causes responses that counteract the change in order to bring back the system to the original condition. A classic example there is when the body tries to fight off bacteria, tries to fight off viruses. It's using white blood cells, using antibodies, using things to try to fight off the infection. Positive feedback, whereas it's common in other scenarios, it's not that common when it comes to the body systems. However, one example is that in childbirth, early contractions causes stretching, and that continues until the baby is born. And positive feedback in general is a change that produces a response that just increases the change. Here are some quick examples that occur in animals. Of course, in humans, we're going to discuss it in the next chapter. We're going to discuss it in the context of the endocrine system and the nervous system, which really maintain homeostasis in human beings. But let's quickly take a look at it in terms of the animal system. Birds and mammals are very good at maintaining homeostasis for all the internal conditions. I mean, really, every organism has to maintain homeostasis in one direction or another, but birds and mammals specifically have, they're warm-blooded, which is a very big difference from other types of animals, and therefore they need to be able to maintain a warm internal condition and have developed very complex mechanisms for maintaining homeostasis. Humans, of course, are examples of mammals. Some examples, the rest of the slide is really based on temperatures, because these ectotherms are cold-blooded animals that don't maintain a constant warm temperature inside. Examples like reptiles, amphibians, fish, invertebrates, which means animals without a backbone. And these things don't necessarily rely on internal mechanisms to keep their temperature constant because their temperatures vary, and for them it's no big deal. On the other hand, mammals and birds, which are sometimes known as warm-blooded animals, are also called endotherms. They produce heat by their metabolic reactions inside in order to keep their blood temperature warm. 98.6 degrees is the optimal temperature for a human being, so of course our body does whatever it needs to do to make sure that even on a cold day, our body is able to maintain a temperature of approximately 98.6 degrees. Now let's take a look at a couple of quick multiple choice questions over here. When a woman is in labor, early contractions cause stretching, which continue until the baby is born. That is an example. Well, we just talked about that a couple of minutes ago. That's an example of a positive feedback loop. Something that is the early stage of labor causes something that helps the labor to continue, that helps the labor to increase. Second question, in humans, the body needs to maintain oxygen levels within narrow homeostatic ranges. If the oxygen level drops, the body responds by doing what? Well, let's see. Using negative feedback to return the oxygen level to normal? Well, that looks about right. Using positive feedback to raise the oxygen level, positive, remember, is when you increase the distinction, you increase the disparity, you increase the change. Here you're decreasing the change, you're going back to normal. using negative feedback loops to lower the oxygen level. Well, if the oxygen level is already too low, then you wouldn't want to lower it further. Using positive feedback to further lower, again, that doesn't make sense for the same reason. And setting the lower oxygen level as the new normal, body can't just do that. The body has to keep the same kind of normal as it always did. There are a couple of slides that I'm going to go back to now that I actually missed in the recording earlier. Here are another couple of examples of feedback mechanisms. After eating a large meal of pizza and salad, the blood sugar level rises. In response, the pancreas secretes a hormone called insulin, which acts to lower the blood sugar level back to normal. People that don't have an appropriate level of insulin or can't produce the insulin necessary are sometimes known as diabetics. Diabetes means an inability to control blood sugar level. So once the blood sugar level then drops to a normal level, then you stop. The pancreas stops producing insulin because it is no longer necessary. A positive feedback mechanism, which in this case is a bad thing, of course, is that high blood pressure damages the kidneys and the arterioles, basically the veins and arteries, things like that, and that causes the release of molecules which further raise the blood pressure, which is, of course, a bad example, a negative example of positive feedback. This example is just a very basic just a couple of diagrams that really illustrate the same example, one example of negative feedback and one example of positive feedback over here. Now for the rest of the course, we are going to be looking at the various human systems. And we're going to start with the endocrine system. Now the endocrine system is a little bit more loosely defined than various other systems in the body, whereas in other parts of the body, organs that are part of the same system are very close together. In the endocrine system, it doesn't really work like that. You kind of have different glands in different parts of the body. All the way up from the hypothalamus and the pituitary gland, which is way up here. It's the thyroid gland, all the way down to the pancreas, and various other glands throughout the body. So it's not really so much as, a system, but it is a system, but it's not contiguous. They're not all together. Rather, the endocrine system is just a bunch of different, glands throughout the body. And what these glands do is they cause what are called secretions, or hormones, that are used to maintain homeostasis throughout the body, to kind of either counteract negative forces or increases positive forces that occur in the body. So the way these things work is that all of these organs, all of these endocrine organs, have to communicate with each other. And they can communicate directly if they're next to each other, for example. They can communicate through neurotransmitters, which sends messages. Later on in Lesson 13, we will discuss the brain and the nervous system. But basically, the brain and the various neurons that go all over the body send messages to the various endocrine glands that allow them to create whatever hormones are necessary. The body also sometimes actually sends messages throughout the body by using secretions. The pituitary gland, for example, which is all the way on top, the main organ all the way on top of the endocrine system, the way it operates and the way it sends messages throughout the system is it secretes various hormones, it secretes various secretions, and those actually tell the other endocrine glands what to do. So this is kind of a review of the previous slide. The chemical messages are sent by the endocrine system either through hormones, which are endocrine secretions that go throughout the body in the bloodstream. And then when these secretions that are created by the thyroid or created by the pituitary gland reach whatever other glands there are, like the liver and the pancreas and things like that, they can then produce things in response to whatever messages they get. And endocrine systems also are controlled through neurotransmitters, which are nerves, which are controlled by the brain, goes down through the spinal cord, and spreads out throughout the body. These are some examples of hormones that are in the body. There are a whole bunch of examples, but here are just three more of the more common and more important types of hormones. Hormones really just means any kind of secretion that an endocrine gland spits out or produces. And here are some examples. You have the prostaglandins, which have different roles throughout the body. In fact, there are many different types of these glands. And I have a little chart over here. You don't have to remember these, but I have a little chart over here of some of the functions of different types of prostaglandins, and you can see that there are many different diverse roles that they play. For example, bronchoconstriction is a constriction of the bronchial tubes. The GI tract is the gastrointestinal tract. That's the tract that your food gets digested. A lot of these have to do with digesting food, and various lipolysis inhibition, lipolysis inhibition, which is the breaking down of fats. There are just many different functions that these types of glands make sure run smoothly. You have cortisol, which is a gland that reacts to stress or anxiety. It actually what cortisol does is it increases your glucose metabolism, which means it increases your breaking down of sugar. It increases your energy. That's why sometimes, if you notice, if you're under a great deal of stress, if something is very dangerous, people very often, their mind becomes sharper, their muscles become stronger, and in general, they're able to get themselves out of situations because they hear stories about people lifting cars and to save themselves or to save their children, and that's because it doesn't always happen that way, but a release of this kind of hormone, of cortisol, stimulates the body to produce more energy and produce more strength on a temporary level. You don't want to have it too much because it's not good for you in the long run, but a little bit of cortisol certainly helps the body react to stressful situations. You also have growth hormone. You can take, and growth hormone acts on nearly all the body cells by causing the body to grow in areas that it needs to grow. All of these types of hormones, by the way, you can take it, and they can produce many of these in the lab, so that if you need more of these, you can just take pills. There's something called HGH, called human growth hormone. People sometimes take that as a performance-enhancing drug. Like professional athletic leagues very often prohibit the use of HGH because it's considered cheating. It's considered an unfair advantage. People take it in order to become stronger. Sometimes people who are too small, whatever reason they're not developing properly, like children, very often they'll take growth hormones in order to try to make sure that they grow on a pace faster than they otherwise would. Here's an example. Here's just a list of some of the parts of the endocrine system. As we discussed before, you have the pituitary gland way up here, which is on the base of the brain, actually, which kind of controls the whole system. You have the thyroid and the parathyroid gland, which is which is near the throat. You have the adrenal gland, which produces adrenaline, something that gives you energy as well. You have the pancreas, and you have various other hormones, glands that are located throughout the body. Okay, let us move on now to two other huge important systems in the body, obviously, circulation and respiration. The circulatory system is composed of the heart and the various blood vessels, arteries, veins, and capillaries. Arteries and veins are the big blood vessels, and capillaries are the small blood vessels. The central component of the circulatory system is, of course, the heart. We, as human beings, have 4 chambers in our heart, 2 ventricles and 2 atria. The atria are in the upper left and upper right-hand corner, the right atrium and the left atrium. Then you have the ventricles. The difference between those two is that the atria accept the blood, and the ventricles pump them. Basically, the blood comes in from the rest of the body to the heart through one of the atria, and then it goes into the ventricle, and the ventricle then pumps it. It's kind of like a pump. It's hard to describe offhand, but one part of the pump allows the water or whatever it is in, then the other part squeezes it out. There are 2 sides to the heart because one of the sides pumps the blood directly to the lungs to get oxygen, and then the other to the rest of the body. The one that goes to the lung is called the pulmonary, because pulmonary means having to do with the lung. So you can see over here that just in this diagram, you've got blood coming into the pulmonary vein. By the way, the difference between a vein and an artery is that the vein brings blood to the heart, whereas the artery brings blood away from the heart. So let's just quickly take a look at the path of blood through. Let's start up here. And it would go into the heart and the right atrium, then to the right ventricle. Then it gets pumped into the pulmonary artery, which is the artery that goes to the lung. goes up here and then goes eventually to the lung where it gets oxygenated and gets the oxygen supply. Then it comes back around and scoots back into the heart through the pulmonary vein. Again, this is a vein because it's carrying blood to the heart, and it's pulmonary because it's coming from the lung, into the left atrium, then to the left ventricle, then the left ventricle pumps it throughout the body. Notice the left ventricle has to be the strongest part of the heart because it's going to pump the blood all the way throughout the body, whereas the right ventricle is only going to pump it the relatively short distance to the lungs. Okay, so let's look at the parts of the heart. It's made of 4 chambers, as we discussed: the right atrium, right ventricle, left atrium, and left ventricle. And most of the heart is muscle, very strong muscle, because it's got to pump all that blood throughout the body. The left ventricle has the thickest layer of muscle because it has to pump the blood throughout the body, as opposed to the right ventricle, which only has to pump blood to the lungs, which is a relatively short distance away. Then you have the SA node, which is the pacemaker. The pacemaker is the thing that causes the heart to pump properly. You may think, when you hear the word pacemaker, you may think of an artificial pacemaker, people that have heart troubles who are not able to, where their heart, or people that really their hearts are not able to regulate its pumping. You know, pace, you can probably think the word pace means the speed at which, or the rate in terms of time at which something is done. So if the heart, for whatever reason, is not able to time itself properly so that it pumps except, pump, except, pump, except, once every second, second and a half, whatever the case may be. Actually, it's more like once a second. Assuming 60 pumps a minute, it's about one per second. Or even if you're exercising, you have more. You have like 2 pumps per second. Whatever. The point is that the task of the SA node and the pacemaker is to coordinate contractions of the heart. And if a person's heart can't work in itself, you have artificial pacemakers. An artificial pacemaker is a relatively small, I have a little picture over here of an artificial pacemaker. You can see how small it is. It's really only a few inches big. And people wear it inside their body, and it regulates the pumping of the heart. It doesn't actually do the heart pumping, that has to be the body itself, but it regulates when the heart does its own pumping. And the purpose of all this, what the heart's idea is, to try to get blood throughout the body. Now, blood, why is blood necessary? Well, blood does a lot of stuff that you need. What blood is, it's a transport mechanism. It transports oxygen throughout the body. It transports food throughout the body. It transports hormones throughout the body. Remember, secretions made by the thyroid or the pancreas, like insulin, or the liver, or the pituitary gland, need to get around the body some way, and the only way to get there is through the blood. So when the pituitary needs to get a secretion around the blood, it just pumps it into the blood, and the blood then spreads it throughout the body. Blood is made of plasma, which is most of the blood volume, 55 to 60%, which is basically a liquid, watery substance. That's what makes the blood liquidy, because it's made up mostly of plasma. And also you've got red blood cells, white blood cells, and platelets. The red blood cells carry the oxygen, the white blood cells fight off infection, and the platelets are necessary in order to clot the blood. Like when you get a cut, after a couple of minutes, hopefully, if the cut's not too deep, or the cut you're healthy and all that, after a couple of minutes the blood will start to clot, it'll start to congeal, and the bleeding will stop. Sometimes you need to put a bandage or press it if the cut is too deep, or if there's a big wound, in order to kind of stem the tide while it has an opportunity to clot. And most of these cells, these red blood cells, which is what gives blood its red color, are oxygen-carrying red blood cells. They're called erythrocytes, and how many of them there are, it determines how much oxygen the blood can carry. In fact, there is a hormone, erythropoietin, if I'm pronouncing that correct. But anyway, this regulates and maintains the appropriate number of blood cells. That is caused by it's a hormone, of course. It is produced in the kidney, and it regulates the number of red blood cells there are. Sometimes a person takes an extra amount of this hormone, they can have more red blood cells. Now, if you think about what more red blood cells will do, is it'll give you more energy. Because remember, what do they carry to the cells? They carry oxygen. What does oxygen do? oxygen allows aerobic respiration. And aerobic respiration is obviously necessary and critical for us to produce energy. So it causes a now it of course the counter side, the bad side, is something that you could have levels of oxygen that are too low. That could cause, because there's a loss of blood, if you lose too much blood, that could cause oxygen levels that are too low because you lost too many of these red blood cells. High altitude, when you breathe in air in Denver, which is 5,000 feet up, or on Pikes Peak, which is 14,000 feet up, or on the top of Everest, which is 30,000 feet up, the higher you go, the less oxygen there is in the air, the less you get with each breath. If you're on the top of Pikes Peak or something, you're 14,000 feet up, you might find yourself panting, breathing in a little bit more deeply, or a little bit more tired, and the reason there's just less oxygen. So there's less oxygen that gets into your blood, and therefore less oxygen that your body can use, and you're going to have a little bit less energy. If you're too high, like on the top of Everest, most people wouldn't be able to survive. In fact, pretty much nobody would be able to survive unless you have some sort of an oxygen mask or an artificial supply of oxygen. This could cause an insufficient amount of hemoglobin. I didn't mention this before, but hemoglobin is the substance that carries the blood that is present in the red blood cells. So hemoglobin is a necessary component of the red blood cells. In fact, it's really the whole point of the red blood cells. Conditions like respiratory problems, inability to breathe properly, or heart failure, of course, could impact the ability to maintain the appropriate level of oxygen. Another disease is called anemia, like sickle cell anemia or different types of anemia, that's caused by an inadequate number of erythrocytes, in other words, an inadequate number of red blood cells that are caused by a variety of different factors. And, as you can imagine, one big symptom of anemia is weakness. Sometimes people will say, you may have heard the expression, if somebody is very weak, they might say he's anemic today. And the reason why is because anemia means you don't have enough oxygen, which means you're going to have a decrease in energy. In terms of blood, you've got oxygenated blood, which is bright red, and deoxygenated blood, which is dark red, like maroon red. So the difference is that oxygenated blood will be oxygenated. Blood will have oxygen in it if it's coming from the lung. Once it goes to the lung, it gets its oxygen. Heart pumps it throughout the body. After it's been around the body, what's it been doing the whole time? It's been giving out oxygen. It's like a delivery truck. After it's made all its stops and given out all its oxygen on its way back to the heart, well, then it's going to be deoxygenated. That's why when people bleed, they tend to bleed a little bit of a darker red, because when you bleed, very often you're bleeding from a vein. If you pump blood from a vein, and a vein is bringing the blood back to the heart, so it's probably going to be a little bit darker. Contrary to the rumor, blood is not actually bluish, even though your veins are bluish. If you see them through the skin, the blood itself is not blue, even if it has no oxygen. The blood, of course, has to regulate itself, and here's another example of feedback. If you have an oxygen deficiency, you don't have enough oxygen in the blood, then the erythropoietin can be produced by the kidney, which is the hormone that increases the number of red blood cells and hemoglobin. That, of course, produces red blood cells. The red blood cells themselves are produced in the marrow, and they carry around more of these red blood cells with more hemoglobin, with more oxygen, and that causes a restored oxygen level. And then once you have the oxygen level back to normal, then that stops the erythropoietin from being produced anymore, because it is no longer necessary. Very closely related to the heart and the circulatory system is the respiratory system. And the respiratory system essentially includes the lungs and the pipes that get oxygen into the body and carbon dioxide out of the body. Respiration is the process, respiration in the cell, cellular respiration means the production of energy, but when you're talking about on a system-wide level, the respiration is the process by which organisms exchange gas with the environment, and that supports cellular respiration. Cellular respiration needs oxygen to do aerobic respiration, and it also produces carbon dioxide. Well, if the cells are going to need oxygen and produce carbon dioxide, that means the body eventually is going to have to get oxygen in, and it's going to have to release carbon dioxide out. And that process is, of course, called breathing. You breathe oxygen in and put carbon dioxide out. Respiration in humans takes place in the alveoli, which are tiny little air sacs that are in the lung. Here's an example of what the respiratory system in general looks like. You've got the sinuses, or the mouth. As we all know, air you can breathe in or out either through your nose or through your mouth. Then you've got the trachea down here, which is the windpipe, which is the thing that the gas goes in and out of. Carbon dioxide goes up when you breathe out. Oxygen goes down when you breathe in. And then you have the lungs. And lungs are millions and millions and millions of these tiny little air sacs called the alveoli. Each alveolus, which is the singular for alveoli, is kind of like the end of a pipe, the end of the trachea, the end of the bronchial tubes. These things are these little tubes that go all over the place are the bronchioles, or the bronchial tubes. If you have bronchitis, for example, it means it's an inflammation of the bronchial tubes. There's a problem with the lining to your lungs being inflamed, and that's why you cough a lot, because it's very inflamed over there. And these alveoli, they cause gas exchange with the blood capillaries. You can see these purple things over here. These are blood capillaries that wind around the bronchioles and wind around the alveoli, and oxygen and carbon dioxide are exchanged through that. The blood gives all of its carbon dioxide to the alveoli, and the alveoli gives its oxygen to the blood. So when you breathe in, you're taking oxygen down into the alveoli, and that goes into the bloodstream. And then quickly the bloodstream gives away its carbon dioxide to the alveoli, and then you breathe out, and the carbon dioxide in the alveoli gets expelled into the environment through your mouth or nose. All different organisms have different types of respiratory structures, and they are required to facilitate the diffusion of gases, which means the exchange of gases between the organism and the environment. Most of these are moist, like your mouth. Well, obviously your mouth is moist, but the bronchial tubes in general are moist. In some ways, that even exposes them to infection because of the fact that bacteria like to live in warm, moist places, and your bronchial tubes are warm, moist places. So the respiratory surfaces are thin to minimize diffusion differences. In other words, to allow easy diffusion back and forth, to allow gas exchange relatively easily. And they have a large surface area. I mean, think about the lungs. Think about that each of these little tubes ends in millions of these alveoli over here. You can imagine the amount of surface area that's in your lungs. The amount of these alveoli, if you counted millions of these, and they're round and shaped like bubbles like that, that's really a lot of surface area, because that's really necessary in order to get enough surface area to transport the appropriate gases to and from the blood. Skin of an earthworm. You know, some earthworms actually breathe through their skin. We don't do that. You know, fish breathe through their gills, trachea, and humans and other mammals use these alveoli, which I showed you a picture of a moment ago. Breathing in is done through inhalation and exhalation. Breathe in, that's inhalation. Breathe out, that's exhalation. In inhalation, the chest cavity is enlarged, and the diaphragm, which is the muscle underneath the lungs, you can see in this diagram, which I'll put back up over here, this is the diaphragm down here. And when the diaphragm relaxes, it moves back up and pushes the lungs so that the air is driven out of the body. And when the diaphragm contracts, it moves down, and therefore there's more space for the lungs. Think of the lungs like a balloon. When the diaphragm relaxes, it moves up and squishes the balloon a little bit, moving the air out. And when it contracts, these muscles down here near the ribs contract, drawing the diaphragm down, causing the lungs to expand. Okay, I found you a little moving diagram over here that illustrates this point, from Wikipedia, of course. This is inhale, exhale, inhale, exhale, because the muscles that control the diaphragm are down here. So when they contract and they pull, the system goes down and you breathe in, because these balloons, these lungs, get bigger. And then when they relax and they contract, they go back up, and that pushes the balloon smaller and gets rid of the air that is in your lungs. Okay, this we discussed before a little bit. This is the gas exchange. This is the heart, of course, in the middle. This is the lungs and the alveoli that are over here. And this is the rest of the body. So, of course, the lungs have to pump the blood to the heart, where it picks up all this oxygen and gives away all this carbon dioxide. Then it comes back to the heart, and the heart then pumps it to the rest of the body. So remember, in the right atrium, up the right ventricle to the lungs, getting rid of the carbon dioxide and taking in the oxygen from the alveoli. Back to the heart in the left atrium, then to the left ventricle, and then back to the body, where it gives away its oxygen and takes in the carbon dioxide. The brain, of course, has to control the heart and has to control the entire respiratory system. The medulla is the part of the brain that is the respiratory control center. Brain damage the medulla is the lowest part of the brain, by the way. We're going to learn it later. You have the cerebrum, the cerebellum, and the medulla. The medulla controls involuntary activities like breathing. Now, breathing is considered an involuntary activity, even though you could control yourself. You could stop yourself from breathing, but if you're asleep or you're not paying attention, you still automatically breathe. So therefore, it is considered involuntary. So, and if a brain could become damaged, that could cause irregular breathing. Even though the lungs are fine and the heart is fine, trachea is fine, bronchial tubes are fine, everything is fine, brain damage could cause irregular breathing because the brain is the thing that tells the diaphragm muscles when to contract and when to expand, and if the breathing is not done properly, then it could mess up the system, even if all the parts themselves are okay. And as we discussed before, when a person runs, you'll start gasping for breath or breathing faster because you've reached your endurance level and the body needs more oxygen. The body's essentially saying, get us more oxygen, get us more oxygen, get us more oxygen, and get rid of carbon dioxide. Remember, when you run, you respirate faster. When you respirate faster, you produce more carbon dioxide and you use up more oxygen. Okay, let's take a look at a couple of multiple choice questions. Josh requires a heart transplant because of a significant amount of damage. Until he's available, he's connected to an artificial pacemaker, which mimics and replaces the SN node, sinoatrial node. That is the natural pacemaker that the artificial pacemaker is designed to replicate. So we'll say B for that. All of the following are respiratory surfaces except, well, trachea of a fly, of course, trachea is a gas exchange mechanism. Gills of a fish, gills are used by fish to breathe. Skin of an earthworm, as we discussed, the earthworm does breathe through its skin. Bat wings, bats are mammals, which means they have lungs just like we do, so that's out. So that D looks like the correct answer. And of course, alveoli of a lion are are mammal alveoli just like human alveoli, so they're also respiratory surfaces. Lesson 11 is nutrition, digestion, and the urinary system. Nutrients, which is the first part of nutrition and digestion, of course, consist of anything that you eat from food or water that gives something that is necessary for humans or animals to survive. There are 6 basic categories of nutrients. The first 3 give you energy. The last 3 are also necessary but don't give you energy. The first 3 being carbohydrates, which are like starches, sugars, things like that, basically things that can be broken down into glucose. Lipids are fats, which give you a very high energy per gram ratio, 9 calories per gram, as opposed to carbohydrates and proteins, which are only about 4 calories per gram. And then you have proteins. Proteins also give you energy, but they also are necessary for other life processes because they are formed by amino acids and they help the body make the other proteins that it needs. Vitamins and minerals are also necessary for the body for various other tasks, things like conducting impulses throughout the body, allowing the body to build certain things. They don't give you energy, but of course they're necessary to survive for various reasons. And water. Water, of course, is essential and often contains dissolved minerals, which you need for your body as well. Water is essential for many purposes, including the fact that most of your body is made up of water. Now, a calorie, which is what measures the energy in food, is not really a food measurement at all. A calorie is actually a unit of energy, or more precisely, a unit of heat. A calorie means the amount of energy required to raise 1 gram of, or to heat 1 gram of water by 1 degree Celsius. If something cools down by 1 gram of water cools down by 1 degree Celsius, well, then it has lost a calorie. If it heats up, it's gained a calorie. The energy contained in food is essentially the food gives your body enough energy to produce that number of calories of heat. And a food calorie, by the way, is also 1,000 calories. So 1 calorie that you eat doesn't actually give your body enough energy to raise 1 gram of water by 1 degree Celsius. It really gives your body enough energy to heat 1,000 grams of water by 1 degree Celsius, or 1 gram of water by 1,000 degrees Celsius. Of course, your body never gets that hot, hopefully. But the point is, so a food calorie is a lot bigger than regular calorie. When you have a piece of bread that says it has 80 calories, it doesn't really have 80 calories. It really has 80,000 calories. But that's just the way we measure it. 1 calorie is really such a small amount of energy that it really wouldn't help us very much. So that's why they're sometimes called kilocalories if you look at on labels, and sometimes, or they could be used calories with a capital C. Carbohydrates are sources of quick energy. Those include things like glucose, which is sugar, or sucrose, fructose. These are all things that are also sugar. Then there are polysaccharides, which essentially are starches, which are long sugar molecule chains, and the body, of course, can use hydrolysis to break them apart into individual sugars. If the body has enough carbohydrate and the body has enough and it doesn't need a particular piece of food that you eat, and so what the body does is the body converts it into glycogen. And glycogen is stored in a manner that it can be used quickly for energy. So athletes, people who work out, sometimes carbo load, like you eat a lot of spaghetti or bread or chips or whatever it is before a big workout or before a marathon or something, because that causes the body to store a lot of it in the form of glycogen, where it's very easily convertible into energy when you actually need it. Then you've got vitamins. Vitamins are molecules that cannot synthesize. In other words, they can't produce other things. They can't reproduce themselves. They can't give you energy, but they are necessary for cell growth, function, and development. Vitamins can be water soluble, which means vitamins that do dissolve in water. And if that's the case, they're constantly being dissolved in your blood, in the fluid in your body, and constantly being drained out and released from your body in your urine, and therefore they don't stay in the body for a long time, and they need to be eaten constantly. Something that is fat soluble, which is stored in fat, that can be held by the body over a much longer period of time. And sometimes even a low-fat diet, because these things get stored in fat, they get put in fat and they're part of the fat molecules. Not having enough fat can actually cause a deficiency in those diets. Of course, thankfully, most of us are in a position where we don't really have to worry so much about not having enough fat, but that's another issue. Here are some examples of water-soluble vitamins and fat-soluble vitamins. Again, you don't have to memorize this list, but this is just some examples. The B vitamins, vitamin C, folic acid, niacin, and biotin are different types of water-soluble important vitamins for the body. Fat-soluble vitamins include vitamin A, vitamin D, vitamin E, which is an antioxidant, helps promote heart health and various other wonderful things for the body, and vitamin K, which is important for proper blood clotting. Looking at this list, by the way, in case you're wondering, I just paused it so that I could take my vitamin D pills. Very often people that live in these cold weather places and don't spend enough time outside tend to find themselves with vitamin D deficiencies. Many of you listening probably also have vitamin D deficiencies, and so sometimes doctors tell you to take pills that make up for the lack of vitamin D that we get. Vitamin D actually comes from sunlight, in addition to some of the foods that we eat. And so if we don't get enough sunlight or don't get enough vitamin D, we can take pills. Now let us move on to digestion. Here's a look at the digestive system, by the way. Sometimes I like to show you kind of a view of the digestive system before we actually look at the various parts. The digestive system starts all the way up in your mouth and ends in your rectum over here and the large intestine over here. Essentially, when you eat or drink whatever food it is, this is the mouth, of course, and then it goes through the esophagus, which is the food pipe, and then this over here is your liver, which secretes something called bile, which helps break down fats. And it actually doesn't go into the liver, it goes straight into the stomach. The stomach has acid called gastric juice, which is made partially of hydrochloric acid, which breaks down your food. Then it goes into the small intestine, which is this big thing which is this long, windy tube over here. And while in here, the nutrients that we do need to fuse into the bloodstream. The nutrients that we don't need eventually end up in the large intestine, where they move around. The water gets absorbed back into the body, and eventually it leaves the body as feces. All right, so let's take a look at the digestive system. Digestion means the process that physically grinds up food and chemically breaks it down. There are really two different types of digestion. There is physical digestion, to physically break down the food, that starts all the way up in the teeth. Then there's chemical digestion, which chemically breaks down the food. It starts in the saliva, and it ends in the small intestine, and does lots of stuff in between. The animal digestive system, such as the human one, has a series of compartments in which food is processed, and various organs that aid in digestion, like our liver, like our pancreas, which creates insulin to break down sugar. I think we discussed that previously as well. There are some other, in this one you have the liver. The gallbladder, which is in the liver, stores bile, which breaks down fats, and it releases that into the top of the small intestine, like over here. When the food comes out over here, there's actually a little area which you don't see in this diagram at the beginning of the small intestine called the duodenum, and that's where the gallbladder secretes the bile into, and that's probably around this area over here, and that's where the fats get broken down. Carbohydrates actually start getting broken down by the saliva in the mouth, whereas meat and fats get broken down by the acid in the stomach and by the secretions in the small intestine. Animals who eat the bodies of other animals, such as, for example, meat, and plants, and vegetables, things like that, some of these are fairly difficult to digest. And so, therefore, we need a lot of help, or we need a lot of secretions and acid and whatnot to break down pretty much everything that we eat. So, here are some of the examples of secretions that help in our digestion. Our mouth contains salivary glands, and salivary glands produces, of course, saliva, which is sometimes known as salivary amylase, which is essentially the same thing as saliva. What they do is they break down starches. If you might not want to try this because it's kind of disgusting, but if you take a cookie or a cracker and you chew it up in your mouth, and you take it out of your mouth and you look at it, it's going to have a much different consistency than it did. If you let it mix with the saliva for a minute or two and then you take it out of your mouth, it's going to have a much different consistency than it did before you went into your mouth, because part of it is already being digested. You also have the stomach and the mucus, and if you chew starches long enough, they'll begin to taste sweet. I don't think I've ever actually tried that. Maybe I'll try that. Take a cracker and keep chewing it up and wait until my saliva breaks it down into simple sugars, but maybe it would work. There's also mucus and water, of course, in your food pipe, in your esophagus. The stomach lining itself contains gastric juices, which contain pepsin and hydrochloric acid, which help break down the foods that you eat. And the cell lining in the stomach produces mucus that protects the walls of the stomach and the other internal organs. The small intestine produces many other secretions as well. Okay, and now we're going to move on to what happens, with the stuff that your body doesn't need. Well, as I mentioned before, the solid food that your body doesn't need eventually gets released by feces over here. This is the rectum area over here at the bottom of the small intestine. But the other part, urine, is the other metabolic wastes produced by your body. This is the waste that's produced in your bloodstream. Your bloodstream does all sorts of stuff, and there are waste products. And the waste products are filtered out by the kidney. All the blood in your body at one point or another runs through the kidney. The kidney's part of the track. Just like all the blood in your body has to run through the lungs to get oxygen, they also have to run through the kidney. And they have to run through the kidney in order to filter out the wastes, the salts, the stuff that your body does not need. And urine is this watery fluid that contains substances that have been filtered out by the blood. So the role of the urinary system, of course, is to maintain homeostasis by excreting waste caused by the cell. Cellular wastes are waste produced by the mechanisms in the cell. The cell produces stuff, and it has salts and other things that are left over. Now, the basic unit of the kidney is the nephron. The nephron, I have a little diagram over here of it. This is the kidney on the left side, which is the major excretory organ, the thing that produces the urine. Then you have the nephron on the right side. The nephron is to the kidney as the alveoli are to the lungs. Just like lungs consist of millions of alveoli, the kidney consists of millions of nephrons. And the nephron is really a filter. The nephron's got these blood capillaries, just like the alveoli. Again, a comparison, the alveoli has blood capillaries wrapping around it to have the gas exchange. Well, the nephron gets the blood from the blood capillaries and then filters out the salts, and then eventually the clean blood returns back into the capillaries. So the nephrons are, well, the kidneys are a paired organ at the waist level on the side of the spinal column. You can feel where your kidneys are. You put your hands along your sides and you feel where the bones are. Those bones, the pelvic bones, are there to protect the kidneys. If you notice in general, you have bones basically protecting all of the most vital organs in your body. Your heart, your lungs are protected by your rib cage, your brain is protected by your cranium, and your pelvic bones protect your kidneys. And the kidney has about 1 million nephrons, so that's a lot of nephrons. And the way the nephron works, essentially, it's got a Bowman's capsule at the closed end of the nephron over here. This is the Bowman's capsule way out on top over here. And from there, you have a network within the Bowman's capsule, and that filters out the fluid from the blood capillaries. And, you know, it's not really critical that you remember all these specific names over here. Just remember the basic idea in terms of how this works. You got the blood capillaries over here, and they filter the blood into the Bowman's capsule, filters out the salts, filters out whatever needs to be gotten rid of by the body, and then it goes back into the bloodstream. And then, eventually, once all this stuff is filtered out, the salts get mixed with water, and they become urine, and then the body gets rid of it. And I guess you probably know how that happens. All right, so kidneys regulate body water content also. The more water your body has, if your body has excess water and needs to get rid of it, you go to the bathroom more. On the other hand, if your body doesn't have enough water, you go to the bathroom less. Because there is an antidiuretic hormone, which is a hormone that decreases urine production. So therefore, a person who doesn't have water, a person who goes many hours without drinking any water, will have a higher blood level of ADH, which increases the antidiuretic hormone, which decreases the production of urine. That, by the way, is when people are when people are dehydrated, their urine tends to be much more yellowish, whereas when people are drinking a lot and they got a lot of fluid, their urine tends to be more white or less yellow. And the reason is because when you don't have a lot of water in your body, the body stops producing urine. The body says, hey, we need that water. Don't get rid of it so quickly. And so the urine that it does produce is much slower and contains much more of these salts and these things that turn the urine yellow. So excess caffeine intake decreases the amount of ADH, which causes increased urination, which can lead to dehydration. That's one reason, caffeine, you drink a lot, it tends to make you go to the bathroom, and so you have to make sure, if you want to not dehydrate yourself, if you want to drink plenty of fluids, don't drink a lot of highly caffeinated fluids, because the highly caffeinated fluids are not going to help in the long run with dehydration. You're better off drinking something without caffeine. Kidneys also regulate oxygen levels with erythropoietin, which we saw earlier when we were talking about the circulatory system. It's released by the kidney in response to reduced oxygen levels, and they increase the number of oxygen-transporting red blood cells. That helps you transport more oxygen throughout the body when needed, and therefore athletes sometimes take a form of this hormone for the same reason, to increase the number of red blood cells, which increases their energy capacity. In invertebrates, I'm not even going to focus much on this, but invertebrates like sponges and small little animals, not really focused on in this course, still what they do is they just kind of get rid of their waste in a much less complicated manner. They don't have such complicated things like kidneys and nephrons and whatnot. And now let's look at some multiple choice. Samantha woke up late this morning and left home without any breakfast. She realized that she was late and began sprinting to the lecture hall 2 blocks away. Which of the following is a likely source for her activity? Well, she didn't eat any sugar or anything that morning, so she has to get it from a stored source of fat. So that stored source of fat, as we discussed at the beginning of the chapter, is glycogen. When you eat carbohydrates and your body doesn't use it right away, it stores it as glycogen, and therefore that's the thing that her body would be using to get the energy. A man survives on a plane crash and ends up in the desert for 2 days with no water. So what is going to happen to his ADH level? His antidiuretic hormone level. Well, think about it. The body isn't getting any liquid and it isn't getting any water, so it wants to conserve the water. In order to conserve the water, it wants to stop the urinary process, the urinary making process. Antidiuretic stops the urinary process. So it probably means the body is going to have a higher than usual ADH level because the body is producing more of this ADH in order to stop the body from urinating in order to conserve whatever water there is. So we'll make that C. Now we're going to look at defenses against diseases. Now that, of course, is really just the same thing as maintaining homeostasis. A disease is a disruption in homeostasis, and defending against disease is trying to maintain homeostasis. How do people get diseases? Well, very often they get diseases from microbes. Microbes are colloquially known as germs, which are microscopic living organisms, including bacteria, which cause things like strep throats, protists, which cause other diseases, fungi, cause things like athlete's foot, and viruses, which some scientists don't consider living things, but whether they're living or not, they can certainly make people very sick. Pathogen is a germ or a microbe that uses another organism as a host, really a parasitic organism. And some examples include that pathogens cause strep throats, colds, coughs, bronchitis, flu, cholera, chickenpox. Virtually any contagious disease is caused by pathogens. How does the body defend against that? Well, the bodies have immune systems, and the immune systems try to defend against these pathogens. One, non-specific external barriers, things that try to prevent microbes from entering the body. Things like skin even, doesn't say over here, but tears, saliva, mucus, these are things that kind of try to block the microbes from coming into the body. Then there are immune responders that try to engulf or destroy them. Things like the white blood cells. Inflammation, fever, are natural killer cells. These are things that are caused when your white blood cells, when your immune system is trying to fight off the pathogens. It causes an increase in energy level of the body, and that's what causes a fever because it just causes everything to heat up because there's so much more activity in there. Then you have specific internal barriers, which is the adaptive immune system. The adaptive immune system is the sort of thing the body learns to fight. When you get a particular type of disease, a particular type of virus, the body then develops antibodies in order to fight it off next time around. That's really the way vaccines work. First you get, and we'll discuss this a little bit later on as well, but when you put a disease in the body, that teaches the body to fight it off next time it comes around. The immunity cells the immunity system, the system that your body uses to fight against pathogens, includes white blood cells, lymphocytes, which are specialized white blood cells. In the adaptive immune system, you have B cells and T cells, and what those do is they help develop antibodies which fight off specific foreign invaders. Tissues and organs that are important to the immunity system include bone marrow. That's where blood cells are created. You may remember when we discussed the blood, the bone marrow makes the red blood cells, while bone marrow makes the white blood cells too. That's, of course, one of the reasons why bone marrow is so important. The lymphatic system, which includes the lymph nodes, which have macrophages, which are things that kill germs, and lymphocytes, which also kill germs, aligning the narrow passages where the lymph flows in between the cells. The thymus is located slightly above the heart, which develops T cells. You could call it one of the endocrine system. It's like here in the body. You can see here's the thymus. The thymus is, it looks kind of in between the lungs. The heart would be somewhere around here, so it's kind of pretty close to the heart and in between the lungs. You also have the spleen, which is a little bit lower in the body. The spleen is a little bit closer to the stomach. Here's a little diagram of where the spleen is over here. You can see here's the stomach and here's the spleen. And that filters the blood. And there are also some other tissues in the body that are important to keep germs out, including your tonsils, leukocytes, and things like that. Leukocytes are white blood cells that specifically attack and destroy invading cells. And they even attack the body's own cells if they become a danger for whatever reason. One group of leukocytes are called phagocytes. Phagocytes are basically any type of cell that surrounds and destroys another cell. Really, something like when a cell tries to capture something by surround it, is sometimes referred to as phagocytosis. So phagocytes are kind of white blood cells that kill other cells or kill viruses or bacteria by surrounding it and crushing it. Inflammatory response is when the body uses inflammation as a defense mechanism. If you have a wound with tissue damage, so the white blood cells and the defense mechanisms of the body inflame the area which blocks it so that it cannot be easily invaded by germs. A fever, and this we discussed already, if a population of microbes succeeds in establishing a major infection, the body might produce a fever. One of the things, by the way, a fever does is it increases the body temperature and causes the invaders to be able to function less efficiently. Then there are the specific disease-fighting cells. Disease-fighting cells include T cells, which we discussed are lymphocytes, cytotoxic T cells, which are cells that destroy infected body cells or destroy cancerous cells. And then there are memory T cells. Again, I'm running through this because I don't want you to try to memorize every single word over here, but just the concepts over here. These are things that you develop in order to fight off future infections. So, for example, if you're stung by a bee for the first time, redness and swelling may occur at the site. And the reason for that is, obviously, the body is inflaming, trying to make sure to prevent foreign invaders from coming in. If you have another reaction that occurs more rapidly, it means the body has learned what the chemicals that come in from the bee sting are and how to react to it. So now let's segue to until now we've been discussing what your body does about foreign invaders that threaten to cause problems. Well, now what can you do from outside your body? What can you take to help your body fight off invaders? One thing you can do is take medicine, which can be good in some ways because it decreases pain and can fight off the infection. But the negative is that sometimes it doesn't let your body do what's necessary to fight off the underlying cause. For example, an aspirin is taken to reduce fever. But remember, sometimes the body intentionally causes a fever by increasing the activity of blood cells and to slow down the viruses. If you kill off your own fever, again, I'm not saying that you shouldn't take aspirin. I'm not saying you shouldn't take Advil or anything like that. But for a minor thing like we have, like a cold or a strep throat or something, is probably not that big a deal anyway. Our body in the long run is probably not going to have much of a tough time fighting it off. But always squashing a fever, always making sure to knock down a fever, might actually hamper your body's ability to fight off the infection. Also, anti-inflammatory drugs can be very good for sports injuries. I've taken anti-inflammatory drugs when I had a back problem, but that's neither here nor there. Inflammation could hamper your ability to function properly. It could be very painful, and it could make it difficult to play sports and do other activities. And anti-inflammatories can prevent the inflammation. But remember, sometimes the body specifically inflames the part of the body in order to prevent germs from entering. And if you take anti-inflammatory drugs, that could kind of increase the risk of contracting infections because the inflammation is no longer there. You could also try to acquire immunity through something called a vaccine. Vaccines are applicable today, or available today for many, many different types of diseases. Measles, mumps, we all take. Chickenpox, most people get. Smallpox, we don't get the vaccine for anymore, but that's because it was destroyed by vaccines. Smallpox was a very deadly disease back in the 1800s also as well, where people would die from it very often. And eventually they discovered a vaccine, and everybody took it, and the smallpox eventually died out. Now they still have smallpox in a lab in a few places, but it's essentially died out from our population to the point where we don't even bother taking a smallpox vaccine anymore. So, interestingly enough, some of the issues regarding when the European settlers came over, one of the weapons they used against the Native Americans, against the American Indians, was they gave them blankets which were laden with smallpox virus. Now, the Europeans were immune from the smallpox virus because, well, this was before the days of the vaccine, but maybe they had gotten smallpox before, or they weren't as susceptible to it. However, the Native Americans, who had never dealt with it, the body had never acquired the ability to protect itself, the body had never learned how to defend itself against these viruses, were much, much more susceptible to this. However, the problem with immunity is that immunity through a vaccine or through getting a disease, like when you get chickenpox, usually you don't get it again. It's possible to get it twice, but not very likely. And there are other diseases like that also. Smallpox was like that also. If a person got smallpox once, they didn't usually get smallpox again. Which is one of the ways they got to the... I don't want to go through the whole story of how they... well, I'll tell you, why not? When smallpox, which was a deadly disease, a scientist named Edward Jenner, back in the 1700s in England, made an observation that the people who were working with cows didn't get smallpox, and he postulated that the people who worked with cows got a different disease called cowpox, which was relatively harmless, didn't kill anybody, just made them sick for a couple of days, and he guessed that was what immunized them, and he turned out to be right. So basically, what he did was he started rubbing cowpox on people and giving them cowpox, and then the body learned to fight off smallpox, and eventually that caused essentially the disappearance of smallpox from human beings. And chickenpox and other vaccines work in a similar manner. The problem is it only works when the disease is very similar. If the disease mutates a little bit, if it's like a different virus, then it doesn't really work. That's why flu vaccines, there's no such thing as the vaccine for a common cold, because there are so many different types of viruses that could cause the common cold. You can't just pick one particular virus and give a vaccine. Same thing is true with the flu. You get a flu shot, but a flu shot may be effective, it may not be effective, because they don't know for sure which flu virus is going to be the one that strikes you. If the same flu virus that you got the vaccine for strikes you, well, then you'll be okay. Otherwise, you might get the flu anyway, even if you got the shot. So the way essentially it works is that the immune system originally doesn't really know how to fight off the virus the first time, and that causes the body to develop the B and T cells that fight it off. Then, the second time you're exposed to that same virus, like the measles or mumps or chickenpox or whatever it is, so then the body already remembers that it needs to produce these particular B cells and T cells to fight it off. It does so much more quickly and more effectively, and you don't get sick in the first place. And you can see here's a little chart that demonstrates how much faster the first time the body reacts the second time around. The first exposure, it may take a week before the body starts fighting it off, and it might take 3 weeks before you get better. And then after many years, you get it again, and now by within the first week, the body has already produced enough B cells and T cells to fight off the whole thing. Remember, T cells are the main thing that fight off the viruses. Now, there are, of course, diseases of the immune system, including allergies. Allergies cause our bodies to have sharp reactions, inflammatory reactions, to things that are ordinarily harmless, like pollen. You take pollen, and normally is totally harmless, floats in the air in the springtime, but if somebody has an allergy to it, then the nasal, the bronchi, the bronchi, the bronchial tubes, the lungs can get inflamed just by exposure to a substance that would otherwise be harmless. There are also various types of autoimmune diseases, which in the immune system gets out of control and attacks the healthy tissue, which obviously is not good. And it could be hereditary, it could be something that you're born with, or it could be acquired. AIDS, which is a famous disease, stands for acquired immunodeficiency syndrome. HIV is human immunodeficiency virus. That's a disease where the body, the immune system doesn't work properly. And if the immune system doesn't work properly, even minor infections like colds and whatnot could be very harmful or even fatal. So here's the human immunodeficiency virus is caused by the AIDS. AIDS actually causes HIV. The acquired immunodeficiency syndrome causes human immunodeficiency virus, and what they do is they undermine the immune system by infecting and destroying T cells, which normally fight off the bad guys, the germs. And there's no effective vaccine against AIDS, but although in today's day and age they do have drugs that keep. In the old days, in the 80s, whatever it was, people used to very often die within a couple of years of getting HIV. Now, very often they have, people can live many years even after contracting the HIV virus. Okay, so let's look at some multiple choice. And the immune cells of Lisa's body have identified some body cells with a specific virus. Which of the following will destroy it? Well, remember, the things that destroy the white blood cells that destroy viruses and destroy foreign invaders are typically the T cells. Cytotoxic T cells, there are other types of T cells, but here the T cells are the ones, or generally speaking, the ones, well, I wrote T, but it's actually choice E over here, because those are the things that destroy the infected cells. Joanne is a kindergarten teacher and makes sure to get a new flu shot. Every single year she has to get a flu shot, as opposed to, chickenpox where you get once in your lifetime. Why is this necessary? Well, the flu is caused by either bacteria or viruses, depending on the year. That's not true. I mean, the flu is always caused by viruses, not bacteria. The shots do not result in the production of memory cells. They do. All vaccines do. Flu viruses mutate rapidly. That's true. They change a lot, so the next time they come around, it might be a different virus causing it, as we discussed. And the antiviral chemicals in the flu shots only last 11 months. That makes no sense because their flu shot is not antiviral chemicals. A flu shot is the virus itself, which gives your body immunity via the vaccination process. Lesson 13 is about the nervous system. The nervous system is, of course, part of the regulatory system. Really, the nervous system and the endocrine system together are responsible for maintaining homeostasis inside the human being. Now, just like all of the human being is made up of different types of cells, muscle cells, fat cells, whatever, the nervous system is also made up of cells. Nerve cells are called neurons, and they are used to transmit electrical impulses for communication. The brain is the center where all the ideas and communications are generated, and then the nervous system has long lines of neurons that transmit the impulses from the brain throughout the body. The tasks of the neurons are to receive, process, and transmit information, and of course to control the movement of all the muscles to which they're attached. The path by which the nerve impulses is the dendrite, the cell body, the axon, and the synapse. We'll see a diagram of that in a minute. The dendrite receives the information, and then the nerve can produce neurotransmitters. Here's just an example. It starts over here. These are the dendrites out here, and the middle over here are the center of the neuron. It's the cell body. Then these lines are the axon, and the gaps, the places between the two neurons. There are three neurons in this picture. This neuron, and this neuron. See, they've all got dendrites. Dendrites are these leaf branch-like appendages at the end, which are specifically designed to be able to accept electrical impulses. That's, it gives them more surface area. Then there's the cell body in the middle. Then there's the axon, which are these long lines between them. And the little gaps between the neurons are called the synapses. And so what this does is that electrical symbols travel from the dendrites and over here and converge into the cell's body over here. And then it processes the information, and the cell body adds up the electrical signals it receives from the dendrites, which may spur positive action or it may spur negative action. It may cause the nerves to say, okay, do XYZ, or make sure XYZ is not done. When the sum of electrical symbols is sufficiently positive, it produces an action. It produces maybe a reflex, it produces a thought that causes us to move our hand, whatever it is. Okay, so the action potential is carried down the axon and then transported. Then the message basically is transported down the axon and across the synapses and to the next neurons, where the process is repeated until it gets to whichever part of the body actually needs to do the stuff. Like when your brain sends a message to your arm saying to lift your arm, like I'm doing right now, although you can't see me, I'm waving, hi. That took a lot of, it didn't seem like it took me a lot of effort, but it took a tremendous number of brain impulses shooting down my spinal cord and then off to the right, into my right arm, into my right hand, causing all this stuff to be done. Caffeine lowers the threshold for an action potential, which means it causes us to be more jumpy. You didn't imagine it. People who drink a lot of coffee really are more jumpy, and that's because there's less of a signal that's required in order to stimulate an action. In order to facilitate the movement of these impulses are the electrical signals that are flying all over the place, the neurons produce a substance called myelin. Myelin, well, the thicker the axon, the faster. It's like if you have a copper wire, and if you want to transmit a lot of electrical impulse over a wire, it's going to need to be a little bit bigger. So the same thing is true with the axons. The axons are kind of like the electrical wires that go into the wall or into the utensil, whatever it is. So when the axon is covered in fatty myelin, the action potential can travel even faster. So the myelin, the sheath is like a shield. The sheath is like something that blocks, and that protects the axon and helps speed up action. So essentially that's what the myelin does. The myelin allows the nerve conduction, which the body produces, which the neuron produces, in order to protect the axons and to allow impulses to move at a faster rate. People with the disease MS or multiple sclerosis develop areas in which the myelin has disintegrated, which means that the impulses that are moving around through the neurons are not going to be able to move around as quickly or as efficiently. And that's why it can result in paralysis or weakness, because the impulses are not going to travel around the body as efficiently as they otherwise would be able to do so. So those are the individual neurons. What about the nervous system in general? Well, the nervous system in general has two basic components. You have the central nervous system, and the central nervous system are simply the key components of the nervous system. Those are the brain and the spinal cord. The brain is where all the thoughts and signals start. The spinal cord is really the thick tumble of neurons that go down the middle of the back of your body. And really, that's like kind of the highway on which everything travels. I mean, think of the brain as being a data center with the computer, and the spinal system as being 100 wires together, and then the wires kind of spread out around the room. So and when they spread out, that's called the peripheral nervous system, which transmits signals from the brain and the spinal cord to the rest of the body. What causes these signals to travel? Neurons. And the neurons that are designed to sense stimuli, to sense things, to get messages, are called sensory neurons. The neurons that cause action are called motor neurons. Let me show you a quick diagram. This is a very, very basic drawing of the person's nervous system. You got the brain over here, the central component of the central nervous system, the spinal cord where virtually all of the brain's impulses are going to go down, then they kind of spread out into the yellow, which is the peripheral nervous system. Now, the motor neurons, as we discussed, also are based on the somatic and the autonomic nervous systems. The somatic nervous system is associated with voluntary actions. When you lift your hand, when you run, when you do something like that, well, of course they have to be done by motor neurons in the peripheral nervous system. And those are considered part of the somatic nervous system. The autonomic nervous system, on the other hand, controls involuntary actions. Things like heart rate, digestion, respiratory rate, perspiration, breathing, swallowing, things that are not completely, some of them may be at least partially voluntary, but things that are not generally considered voluntary. Things like reflexes and these are also things that are controlled by the autonomic nervous system. Reflexes are actually controlled by the backbone themselves. Here are some examples of neurotransmitters. A neurotransmitter essentially is a substance that is secreted in the body that allows impulses to move more easily from place to place. Here are some examples. There's epinephrine. Epinephrine is sometimes used as a drug from outside the body to help to help cardiac patients because it allows greater oxygen exchange through to the individual cells that may be suffering when a person's suffering a heart attack or cardiac problems, and therefore not enough blood is getting around the body. So it's part of the sympathetic nervous system we discussed before. Actually, I didn't discuss it, but sympathetic is the part that prepares the body for stressful or energetic activity. So that's what epinephrine does. It activates organs in the nervous system to compensate when there's some sort of a problem going on. Also sometimes known as adrenaline. That's probably what we know it as better than. It stimulates the body to generate more energy and to move the impulses around more quickly. Endorphins are found in many parts of the brain and spinal cord, and they block the pain sensation and alter mood very often in a positive way. Endorphins very often make people happy, and you can even increase endorphins naturally. I think even something like exercise is known to increase endorphins and improve people's moods in that way. I was just reading that endorphins is sometimes comparable to something like morphine, which morphine is a very strong painkiller, a very strong sedative. Endorphins can have a similar effect, although on a little bit of a lower level. Then there's serotonin, which is found in the brain and the medulla, which is the lower part of the brain, which influences mood and sleep, helps people sleep, hopefully. And there's glutamate, which is a major neurotransmitter in the central nervous system. These are some things that are important within the body, and they also can be brought in from outside the body to kind of help the person. The brain itself, you've got the medulla, which controls this is really the bottom of the brain. Let me get you a diagram of actually there's one right over here. There's the medulla, which is on the bottom over here. This controls involuntary activities like heart rate and whatnot. Pons is just part of this, the back of the brain. Remember, the pituitary gland was the part of the brain that controlled the hypothalamus. I'm sorry, the hypothalamus controlled the pituitary gland, and the pituitary gland used the various secretions to control the rest of the endocrine system. The pons influences sleep and breathing patterns, and then you have the cerebellum. The cerebellum is the part of the brain that controls movement and balance, this yellow thing over here. You got the medulla that controls involuntary activities. You have the cerebellum, which controls balance and motor functions, ability to move your arms around properly, ability to do what you need to do from a motor perspective. And then, well, they contain many other important, it plays a role in various other things, sleep and wakefulness, emotion, movement. But then you have the number one, the biggest part of the brain is the cerebrum. This is the part where you have the thought that comes from. This whole area over here, the senses, the thought, etc. And you've got the cerebral cortex, which is the thin outer layer of each cerebral hemisphere. This is another diagram we have over here, that this is the cerebrum. This whole thing is the cerebrum over here, and you can see there are different hemispheres or different regions of the brain that control different things. Some parts of it may control memory, some part of it may control the five senses, taste, touch, feel, smell, and hear, and thought and voluntary activities. Those are things that are controlled by the cerebrum. So the neurons in the cortex receive and process sensory information from the five senses. They direct voluntary movements. When you decide to do something, that for that thought is formed in your cerebrum. They create memories, allow us to be creative, and to envision the future. For example, I know that if I watch this crash course and study the PowerPoints, I will do well in this final exam. The lobes are called the I don't even know how to pronounce these, so forget these. There are different lobes over here, and these control different functions within the brain. The brain itself is made up of two hemispheres, which means two halves. I mentioned before that there were several hemispheres. Of course, there can only be two hemispheres. Hemi means half, and sphere means circle. Like there can only be a western hemisphere and an eastern hemisphere of the globe. So you have the brain is made up of two hemispheres. The two hemispheres communicate through the corpus callosum, which is should be somewhere around here. And well, okay. Anyway, so discoveries have shown that women have a larger one than men, and therefore have a greater ability to switch from the left side to the right side of the brain when part of the brain is damaged. Now, there's a lot of talk, especially in psychology, about what the left half of the brain takes care of and what the right half of the brain takes care of, and that's all fine and good. But the point is, the entire brain controls voluntary activity. Different tasks are controlled by different parts of the brain. For example, it's possible that brain damage from a stroke is when oxygen gets cut off from the brain, or other injury can leave a person with the ability to comprehend written language but not spoken language, because written language and spoken language might come from different areas of the brain. And if there is brain damage, only the ability to do certain tasks that were controlled by that part of the brain might be affected. So let's take a look at the some multiple choice. It's possible that brain damage from a stroke or other injury could reach a person with the ability to comprehend written without spoken language, as we just discussed. Which statement best explains this? Different aspects of language comprehension are controlled by different cells. Not really. Remember, there are millions of cells in each part of the brain. All aspects of language are the same part of the brain. That wouldn't make any sense, because then if there was a damage to one part, it should damage all skills equally. C, on the other hand, looks pretty good. Different aspects of language comprehension are controlled by different parts of the brain. And by the way, again, like I mentioned earlier, I hope you're all pausing before I give the explanations in order to try to see if you can answer them yourself. I think that would be the best way to do it. So, after an accident, a professional musician has difficulty comprehending what is said and cannot associate instruments he has played with the sound they make. Which part of the brain is likely damaged? This is clearly the creative, sensory, voluntary part of the brain that was damaged over here. It wasn't a motor function; it wasn't an involuntary activity. So the part of the brain that seemingly was damaged was the cerebrum, or the cerebral cortex, which is the main component of the cerebrum. So the best answer would be C. And finally, our last lesson involves the two organ systems that allow our movement and allow our voluntary activity. And those are the muscular system and the skeletal system. First we have the muscular system. Now the muscular system is the thing that allows movement. A muscle, generally speaking, is a tissue composed of cells that are capable of contracting, which means squeezing together, tightening up, which causes movement, thereby moving part of the body. The three types of muscle that we have in human beings and that are in vertebrates in general, animals that have backbones in general, are skeletal muscles, which are attached to bones that allow us to move. There are cardiac muscles, which are heart muscles that allows our heart to contract and pump blood throughout the body. And then there are smooth muscles, which are associated with involuntary activity, such as breathing, moving blood around, etc. Let's take a look at some of the skeletal muscles in general. First are the leg muscles. The leg muscles contract when lifting heavy objects is determined by the frequency of action potential and number of muscle fibers stimulated. You've got millions of these little muscle cells, and what they're attached to is they're attached to your skeleton. They're attached to your bone. The way they move is when they contract, in other words, they squeeze together, it pulls back, and when they relax, it moves back in the other direction. So when you can contract the muscles behind your leg to lift your leg up, and you can relax them to put them back down. The largest motor units in all the human muscles are the leg muscles. Those are our maybe not the strongest. I think they say that the jaw muscles are the strongest muscles, but certainly the largest and the thing that carries the most weight, literally. The leg muscle fibers, there are fast twitch muscle fibers, which are able to move quickly, and these are things that are built up in runners and sprinters. Then there are slow twitch fibers, which have more mitochondria because they need more energy, they need more respiration. Remember, mitochondria does the respiration, and they contract with less power than fast twitch fibers. And these are built up by long-distance runners. Basically, the idea is that both of these are necessary. The slow twitch fibers are built more for long-term consistency, whereas fast twitch muscle fibers, they produce less energy in the long run, but they can move faster in the short run. Then one of the things that people do in order to try to obviously build up, in order to try to be able to move more efficiently, move parts of their body more efficiently, is to build up muscle strength. And when the muscles become stronger by use, they become more resistant to fatigue. People who work out often, people who use their muscles a lot, often require rapid bursts of high energy muscle output because they're working out and they need fast energy, or they need energy very quickly. And so therefore, sometimes they'll take dietary supplements like amino acids, which make more muscle cells. Muscle cells are built out of protein, and so therefore taking more protein supplements, generally speaking, people who are good athletes very often will take protein supplements. And protein supplements, one of the things they do is they allow you to build more muscle cells, and they increase energy, of course, just like any other energy-producing nutrients. Many supplements contain something called creatine, which can add to the phosphate ions to regenerate ATPs when there's a lack of oxygen. Remember that ATPs, adenosine triphosphates, are a key element in respiration and in aerobic, respiration with oxygen, aerobic respiration, and so therefore adding ATP, which is adenosine triphosphate, has a phosphorus group, and so in order to make sure that there's a constant supply of ATPs, creatine can increase the ability of phosphate ions so that the body will have plenty of these to make new ATPs when necessary. The skeleton, or the skeletal system, is the bone structure, the structure that supports the body. The muscles control. The skeletal muscles are the things that control the skeleton, causing the body to change shape. So you have the bones themselves. Then you have the bones are in the bottom over here, which are hard and rigid and contain lots of minerals, including calcium and phosphate. And the thing about the bones, obviously, is that they're strong and they give you structure, but they're not very flexible. So in between the bones is where we have our flexibility. In between the bones we have cartilage, which contains glycoproteins and has elastic fibers, but cartilage is a hard substance it's kind of plasticky. I think, the best example of cartilage, if you want to know what it feels like, is to take your nose, and the thing in between, the thing that separates the two parts of your nose, at least in the front, is cartilage. In the back you got an actual bone, but in the front you got the cartilage. And so it's kind of like, it's hard, but it's plasticky and not quite bone-like. And then you have ligaments. Ligaments are the things that hold the bones together. One bone to another is held by ligaments. And then you have, by the way, the tendons. A tendon, T-E-N-D-O-N, is what holds the muscle to the bone. So these are made up of collagen fibers, and they don't have a significant amount of elastic fibers. Ligaments are not that flexible. So in between two bones, there's not necessarily a tremendous amount of flexibility. It really depends on the bone itself. You have the endoskeleton, which is the internal skeleton found in a vertebrate. In humans, for example, we have an endoskeleton, which means we've got a skeleton inside our body. There are some animals, like insects, that have exoskeletons, which means their skeleton is actually outside their body. So our skeleton, our vertebrate skeleton, in other words, a skeleton found on a vertebrate, which means an animal with a backbone, like a human being, for example, has an axial skeleton, which contains the head, the vertebral column, namely the backbone, the rib cage, And obviously you don't want the axial bones to be weak. And then you have the appendicular skeleton, which means like the side skeleton. The pectoral and pelvic girdles, which are lower in the body, like in the midsection of the body. The forelimbs, which are the arms and legs. And I'm sorry, the arms and hands. Then the hind limbs, which are the legs and feet. If you want a little diagram, here's a little diagram. We've got the whole skeleton here, including you can see the skeleton face over here. The blue is the axial skeleton, and the brown is the appendicular skeleton. As you can see, the blue skeleton is kind of the main skeleton, the central skeleton, and then everything else, the rib cage over here, which protects the heart and the lungs, the spinal cord, the backbone, which protects the spinal cord, and of course the skeleton, which protects the face and the brain. And then you have the appendicular skeleton, which is the arms, legs, pelvis, etc., the rest of the skeleton. Okay. So now let's take a look at some of the parts of the skeletal system. First, we discussed a little bit before the cartilage, which is the hard plasticky material that kind of coats the bones and does some other functions. Some examples of cartilage are the entire skeleton of sharks. Sharks are examples of what are called cartilage fish. Sharks don't have bones. They have cartilage, which obviously can be pretty hard. Cartilage covers the end of bones at the joints, kind of to make sure that the bones don't rub up against each other. In fact, when the cartilage does get worn away and the bones do rub up against each other, which can be very painful since there are nerves there as well, that's sometimes known as arthritis, which is an inflammation of the joints. Arth means joint, and joint means in between the bones. So that's arthritis. So cartilage supports flexible portions of your body, like your ear and your nose. The nose I gave you before as an example. The ear, underneath the skin of the ear, is also cartilage. And it also provides a framework for the respiratory system, the larynx, the trachea, the bronchi in the respiratory system. Cartilage doesn't have blood vessels in and of itself. And therefore, that's why damage to cartilage, which very often causes arthritis, tends to heal very slowly because of the lack of blood vessels that are over there. Lower back injuries sometimes are to cartilage as well. And that's what causes them to heal slowly. There just isn't enough blood there to bring the oxygen that are necessary for the repairing the tissue. Then you have the joints. The joints are parts of the skeleton where the bones meet with each other, located at the elbows, knees, fingers, and they can move. The regular joints, hinge joints, can move forward, or basically they can't move all over the place. They can move from flat to moving in and out. I mean, if you want to know what I mean, just look at your elbow. Your elbow can be flat, where your whole arm is flat from one point to another, or your elbow can move upwards like a hinge. Then you got the ball and socket joints. The ball and socket joints, where the round end of one bone fits into the hollow depression of another. The classic example there is your shoulder. If you hold your shoulder, you realize your arm can't go completely around in 180 degree in a 360, but it has a lot of freedom to move around in your shoulder joint. Then you got the knee joint. The knee joint needs to be a hinge joint, because otherwise there would be decreased stability. If you would be a ball and socket joint, then your knee would be able to move in any direction. You wouldn't have very much stability. It would be very hard to stand up, very hard to balance. And one thing that very often gets injured, actually, the anterior cruciate ligament, the ACL, is one of the major knee joints, and damage to this ligament is very common in athletes because athletes put a tremendous amount of stress in the muscles in their feet and knees and lower bodies. So an ACL tear is very often a sports-related injury, but can also result in other types of injuries. And when a person has a torn ACL, a torn anterior cruciate ligament, it's much more difficult for the skeletal structure to maintain stability. Here are some examples of knee joints over here. And again, you don't have to remember all of these terms, but this is your knee. You've got the fibula over here, which is underneath the bone, the tibia, which is further down, much closer to the ankle. You've got the kneecap over here, the tendon. These are muscles, so the tendon attaches the muscle over here to the bone over here. The cartilage in between in between the bones. Here's a bone, and in between the cartilage covers up the joint. Okay, and finally we've got the issue of bone strength. Bone strength is increased given a moderate with weight-bearing exercises. You lift weights or you do anything that could increase bone strength, just like any other kind of exercise. When you increase resistance on something, that tends to build up the strength. Many people hope they will one day be able to go into space. I mean, well, I don't know if that people hope necessarily, but the idea of being in space, or being anywhere without gravity, which is really just in space, is that if you are in space, like astronauts, they go up to the space station for a few months, or they go to the moon, or whatever it is, or even just for a few days, the muscle and bone mass are no longer being used. When you're on Earth, your muscles and bones are constantly fighting against gravity, and therefore maintaining their strength, or even building your strength. Whereas if you're under weightless conditions, there is no pull of gravity, and so therefore the bones kind of atrophy away. Atrophy means to lose strength because they're not used and not needed. So what astronauts do is they do special weight-bearing exercises each day in order to try to maintain their bone strength. If they don't, then they'll atrophy away, they'll deteriorate away, which will cause a serious problem for them when they get back to Earth. Okay, let's take a look at a couple of multiple choice questions. Again, you can pause it and see if you know the answers. Okay. In a muscle that is resistant to fatigue, such as a marathon runner leg muscle, you would expect if it's resistant to fatigue, it means it can produce all the energy it needs. Now, something that can produce all the energy it needs, you would expect it to have a lot of ATPs. A limited blood supply would cause it to be fatigued. A very few mitochondria would cause it to be fatigued. It wasn't not producing enough energy. An striated appearance wouldn't work either. That's not a normal thing. The best thing would be elevated levels of ATP. What might be a major disadvantage of a ball and socket joint rather than a hinge joint in a human knee? Well, not decreased strength. That's not a problem. The ball and socket joints are just as strong. Reduced ability to move. On the contrary, it would increase the ability to move your lower leg. Decreased joint flexibility. Again, that's not true because it would increase your flexibility, but it would decrease your stability. It would make it more difficult for the body to hold firm, and so therefore D is the best answer over here. Okay, that's basically what we have for the material. I do also at the end want to go over a couple of essay questions and that you might see on the exam from the exam bank. Just in general, you're not going to see these particular questions, but these are some examples. How would you do this one? Explain how unsaturated fat is structurally different from saturated fat. You know, if you want to pause the recording and see how you would answer that, you're welcome to do so. Well, the way I would answer it is that unsaturated fat, first of all, is liquid at room temperature, as opposed to saturated fat is at solid at room temperature. But unsaturated fat doesn't have all the bonds within the lipid molecule bonded to a carbon, whereas saturated fat does have every single free bond is attached to a carbon. Number 2, normally a certain enzyme has a maximum activity at 37 Celsius, but once it's been heated to 100 Celsius and then cooled back down to 37, the enzyme is no longer active. Why? Well, the reason for that is because once it gets up to 100, it could cause a change in shape or a change in structure of that enzyme. And once that enzyme has been changed, like melted or changed form for whatever reason, it can't go back to the lock and key model. Remember, enzymes have to be in a certain shape in order to act on a particular substrate. Number 3, you're a forensic biologist who's been asked to review the files of an old unsolved murder case. The autopsy report's missing a lot of information, but you read that the victim had a thymus gland that was just starting to shrink. Based on the information, what would you conclude about the victim? Well, if you remember, the thymus gland, which is located near the heart and the lungs, was a key organ in the immune system. So it produces T cells. T cells actually are produced and mature in the thymus. So you might conclude that the victim over here had a reduced immunity to pathogens, reduced immunity to outside diseases or other forces outside the body. You might also conclude that the victim had some kind of a disease that caused problems for the victim's immunity system. Or immune system. Okay, so that's about all we have. I think you definitely have enough information to do well on the Introduction to Life Processes exam. If you'd like to contact us, please feel free to do so. There's contact information in the course syllabus, and good luck.